As every year, this year too, we are holding our Astrobiology Conference. And thanks to your interest, it became as a tradition to make this conference. I want to say all of you to welcome to our MBG Türkiye Agenastro 4th International Astrobiology Conference. In this session, our team member Nigora will be hosting for you. Hi, Nigora.
Hello, everyone, and welcome to our 4th Annual International Astrobiology Conference organized by Agenastro, MBG Türkiye community, which I am a member of. My name is Nigora. I am a third-year biology student at Bursa Uludağ University in Turkey. I will be co-hosting this event for you. So now you can sit back, take your snacks, and enjoy the conference. We are taking off. Our today's very first keynote speaker is a professor of geochemistry and planetary sciences at the University of Science and Technology in China. At the same time, he is a research scientist at the Deep Space Exploration Laboratory based in China and a research investigator in Blue Marble Space Institute of Science, which is based in the United States. Today, he will take us to one of the moons of Saturn, Enceladus, and give us a broad information about its water chemistry and implications on habitability. With that being said, ladies and gentlemen, please welcome Professor Jihua Hao. Hello, Professor. How are you doing?
Yes, I'm doing okay. Should I start now or?
Yeah, if you don't have any other information you would like to add about your background, about your works, whenever you're ready, you can start your screen. The stage is yours.
It's my great pleasure to give this presentation. I was surprised by that this astrobiology conference already run for several times, and I received the emails in the past years as well. And I'm really impressed by that the organizers are some early career students. So it's my great pleasure to present today about my work. So today I will share some.
Yeah. Thank you very much. I am removing myself.
Okay. Thank you. Yeah. So today I will present some results which I started working on this actually back to where when I did my postdoc at Lyon, France. It's originally from Europe. My title today is Enceladus Ocean Water Chemistry and the Implications on the Habitability. So I will present some my published work as well as some unpublished ones in this talk. So this is the outline of my talk. I will firstly briefly overview the exploration history of the Enceladus, and then I will focus on the ocean water chemistry as well as the organic stability in the ocean water, and then last I will mention briefly the habitability and we call it the limiting factors. So the Enceladus was firstly discovered by William Herschel and his sister with the telescope they built by hand in 1789. So William Herschel also proposed the infrared. So it's a great scientist, and he discovered the Enceladus. But we back to 20 years ago, we only know that the Enceladus is a very acid ball. So not too much information about this. We only know that it's the 6th largest moon of the Saturn. And we know briefly the radius is 500 kilometers in radius. And it's located in the E ring of the Saturn, which even today, I think this year, people are still publishing papers about the formation of the E ring and its relation with the Enceladus. And we know that because the surface temperature is very cold, it's like a minus 2000 degrees C. So it's also water rich. So the water forms very thick layers of ice on the surface. And it was until the Voyager, which took the close picture, the photo of the Enceladus, and it really showed that the icy moon is not as like we thought. Previously, we thought that this icy moon is very small. It has so little radioactive energy, from the deep, so it must be dead, like our moon. But then the Voyager took the close picture and showed a very smooth surface, with one of the probably the highest reflectivity of the planetary bodies in our solar system. So to be clear that this high reflectivity of the surface mean that it has overall young surface, because otherwise, like our moon, it will be covered by this very high density of impact craters. But with the Enceladus, you can easily see that although the southern atmosphere is covered by a lot of like impact craters, but then the really the southern part of the acid ball is overall very young surface. Also, it has very big difference in the crater density, means that different age, as well as by the photo, you can see this kind of like a river bed structure. It's actually terrains can be high as 1 to 2 kilometers high, and also has some cracks, like you said here. So it basically shows that this acid ball is very, very young in the surface, and has some active structure, like structural topography characteristics. And then NASA launched the Cassini-Huygens probe to really look closely to the Cassini to the Enceladus surface, and it found it's very surprising to find this like called tiger straps on the Enceladus, which is formed these by the folding of the layers, or the eruption from the internals of the Enceladus. And the most shocking information we got from Cassini is that it found the water plume from the south pole of Enceladus. And then later, actually, NASA arranged the flyby through this plume and collect some acid grains and analyzed them to get the composition of this, and showed very surprising results. I will show it later. Also, the Cassini's payload also analyzed some geophysical properties of this whole planet. Here are two examples. One shows thermal anomaly in the south pole. Basically, you can understand this as the temperature difference variation. It basically shows that the southern pole has an overall higher temperature, and also the temperature anomaly, the distribution of the temperature anomaly is very closely related with this kind of tiger strips structure, which means that it should be like some heating resources from inside. Basically, it's a liquid water based on the geophysical modeling. And also, it showed that it has a large mass anomaly at the south pole, which suggests, based on the geophysical modeling, a large regional subsurface ocean, which could be 30 to 40 kilometers in depth. So basically, we based on all of these results, it showed that it must have some liquid water activity inside of the Enceladus. And then, like I said, NASA ordered the Cassini to fly by the plumes and collect the acid grains to analyze the composition from this plume. And one very useful payload is called the ion and neutral mass spectrometer. And it analyzed the gaseous phase and organic compounds in this plume. It showed that it's basically mostly water, which makes sense because the Enceladus is very water rich. But also it has substantial levels of hydrogen. and methane and nitrogen, as well as ammonia and carbon monoxide, carbon dioxide, and possibly, but not definitely, hydrogen sulfide. If you are familiar with this, with the like ecology, it basically shows that it has a very anoxic environment inside because of the high concentration of the hydrogen and the ammonia. Also, it has substantial levels of organic compounds, which could be very, very large in terms of the structure. So after this, after we found this evidence, very surprising evidence, people suggest the Enceladus discoveries actually changed the direction of planetary science from the Mars probably to the icy moon, which becomes the hottest target for the astrobiology study. Actually, I think just this month or last month, European Space Agency just announced that they will want to go to the Enceladus and will mark the Enceladus as their next top priority target for the landing mission. Another very useful payload is called a cosmic dust analyzer. It can analyze. It collected the acid grain from the E-ring and analyzed the salt composition from the in the acid grains, and it showed that it contains high concentrations of cations and salts, such as sodium, potassium, sodium chloride, sodium bicarbonate, sodium carbonate, and silica. So the salt composition is very surprising because our seawater on the Earth has high concentration of sodium and sodium salts. So which the Enceladus Ocean also has the similar composition, but it has also high concentration of sodium bicarbonate and sodium carbonate, which are alkaline salts. We usually use this in our cooking to neutralize the acid, right? And another surprising finding is the silica. Usually the silica will have very low solubility in the water. If you sink your silica-made jewelry into the water, it will not dissolve very quickly or highly, right? But then the Cassini found high concentrations of silica in other particles. And using this silica, it suggests that maybe there are some high some high temperature fluid venting in the deep inside the ocean, because the silica concentration solubility increases rapidly with the temperature increase. So overall, it showed that not only it has the fluid water, but also the water can alter the rock, has a very active water rock interaction below the icy shell, and possibly the hydrothermal activity. So I will, this is all backgrounds. So I will focus on my research in these 2 years in my following talk. The first one is to constrain the inorganic chemistry of the water in the subsurface ocean. And as we said, the Enceladus plume already gave us some idea about the thickness of the ocean, as well as the activity of the hydrothermal fluid, and also the water composition, the major water composition. Sodium chloride and sodium carbonate rich ocean, and also it's a reduced environment. So to really get some more information from this is that we need to know the large environmental properties for this. One is the pH, which is alkaline. Another one is the redox state, which is here as a reducing ocean. But apart from this, from the Cassini data, we actually have very little ideas about other elements. To translate those Cassini data into the water ocean chemistry, people also did some experimental simulation. it is very notable that my collaborator Frank Postberg designed this facility to simulate the impacts of the hypervelocity ice grain from the ocean worlds in the space impact on the mass spec, and which can allow him to translate this data into the real fluid chemistry below the icy shell. But we don't have we have the idea that based on all of this evidence, it's alkaline and reducing and carbon-rich seawater, which is showed as a left figure. Basically, it's close to the Lost City hydrothermal vent, and also the siders the fluid. And but from the Lost City hydrothermal vent, we know that it can support life. But for a better understanding of which kind of life can be found in the Enceladus or can form in the Enceladus ocean, or how much life it can support, we not only need to know this major composition, but also I think more importantly we need to know some minor elements, including the phosphorus, or recently I'm working on the sulfur and the iron and these transition metals. The reason why is that it's like a bucket to hold the water. If you have those abundant elements, it is far from enough to support life. But it will have some very little availability of these very minor elements, whose availability will limit the overall water you can hold in the bucket. It's called the bucket effect. So here I want to focus on the phosphorus, because phosphorus is a critical building blocks of life. It's one of the 6 basic elements to form life. It forms the nucleotides, phospholipids, and materials. Sorry. It is also used as all kinds of life as a very popular metabolism currency. Our human use the ATP, and ADH, FADH, and some microbes use polyphosphate. And for the ecosystem level, on that order, in that schedule, phosphorus availability is a limiting factor for the ocean bioproductivity, which means that the influx of the phosphorus will determine that the overall productivity level of the ocean, because of the poor availability of the phosphorus. Also, recently phosphorus become a hot study topic, because people use the phosphorus as a potential biosignature, especially the phosphine in the Venus story. But before we can understand the phosphorus cycle or the phosphorus availability in the Enceladus ocean, we need to know that which kind of the factors govern the element cycle or availability. Based on our modern Earth, it's basically controlled by the 2 parts. One is the continental weathering, and another one is the recycling in the ocean. Also, it's largely controlled by the precipitation over the secondary minerals in the ocean. However, if we look at into the Enceladus, it's totally different from the Earth. There's no continental land, so you don't need to worry about the continental weathering, which is good. Another one is that it has no evidence of abundant life, so you don't need to worry about the biological effect on the element cycle there, at least for now. And the ocean gets covered by the thick layer of ice, probably for 100 million years or several billion years, which is very good for the modeling, because you basically can treat the system as the almost enclosed system, so that you can allow the the water rock interaction to proceed in a larger extent, or even reach some kind of equilibrium, which is the basic idea of my modeling work. Because we know that from these blue parts, blue characters, are known constraints for these elements, like sodium, potassium, chlorine, total carbonates, and hydrogen and total nitrogen. But we actually don't know about these minor elements like calcium, magnesium, iron, silica, phosphorus, or even sulfur, which you need to make some assumptions. But based on these environmental conditions, we can I think we are very confident that some of this water rock interaction may reach like kind of equilibrium given the very long time in a very closed system. So in my model, I just assume that these elements are controlled by the minimum solubility of the secondary minerals, like for example, calcium by the dolomite or calcite or aragonite, which indicate which means that if there is one mineral has the least solubility, so given a very long time, you will reach this kind of the dissolution and the precipitation equilibrium with this mineral, so that the mineral solubility will control the element. And here we focus on the phosphorus. With the iron and the sulfur, we are currently working on that. Firstly, we need to know that under this very reducing condition, could we have the phosphorus in another redox state? So we basically did the called the Bombay diagram, which shows the pH and kind of EH, but here as the log activity of the dissolved hydrogen as the indicator of the redox state. And this green line, dashed line, shows the inferred insidious ocean water chemistry, like upper limit. Basically, from this figure, you can see that the also phosphate as a phosphate in the fifth wavelength is the dominantly stable valence, even under very reducing conditions relevant to the insidious ocean. And for this reduced phosphorus species, for example, phosphite or phosphine, they are not stable at all. And if there are some way can transport them or they inherited from the original accretion, those species are not stable at all. It could be it would tend to be oxidized to form this also phosphate, given again the very long time of the water rock interaction history. So probably it can be a potential back signature because under reducing conditions, the microbes can form this kind of reduce the phosphorus species based on our Earth systems. And here's the results. So we found that given a very large range of the pH and also a very large range of the boundary conditions like carbon dioxide, ammonia, nitrogen abundance, etc. So we basically have very high solubility of the phosphate, which is orders of magnitude higher than the modern Earth seawater. And we also consider two cases. One is the with chlorine case, another without fluorine case, because based on our daily knowledge, you know that with some fluorine, it can suppress the dissolution of of your teeth, right? That's because your teeth is in the middle of calcium phosphate, and the fluorine can form very strong and very less soluble fluorine apatite. So that's why you will have a little bit lower overall phosphate concentration than this without fluorine case. But nevertheless, both cases show that the phosphorus level in the insidious ocean water could be much higher than the modern seawater, which is very good for life. And we also did some sensitivity tests about our modeling, why we have this high solubility. We particularly interested in the carbonate concentration, and we based on this figure, that it showed that if you increase the carbonate concentration, the predicted phosphorus solubility will increase as a logarithm linear relationship. So basically, with the higher concentration of carbonate, you will have the higher solubility of phosphate, which makes sense because if you have high concentration of the DIC, the dissolved inorganic carbonate, it will suppress the dependent metals because based on the equilibrium with the those carbonate minerals, which can increase the solubility of the phosphorus minerals because the phosphorus can also form the precipitate with the dependent metals. And do we have any ideas about whether this is true or not? Yes, we have some observations based on the modern Earth's alkaline lakes. I'm sure Turkey has some alkaline lakes. So basically, from the alkaline lakes, based on previously like almost half a decade, several decades, we know that the alkaline lake have this logarithmically linear correlation between the dissolved phosphate with the carbonate, and recently, also strengthened by the Toner and the Kathleen in 2019. Basically show that the similar story is like higher levels of dissolved carbonate allow high solubility of the phosphate. And we also did some sensitivity test with the temperature because, as we said, Enceladus ocean may have the active hydrothermal activity. But we show that the results show that temperature has very, very little effect on the phosphorus concentration. And we also did some sensitivity tests with the silica concentration because the silica could be pretty high in concentration in the Enceladus ocean water. Basically, it showed that the silica have some effect, but not very large effect. Also, given those very long water rock interaction history, the amorphous silica is not stable. It may tend to form this kind of less crystalline, like silica minerals, like alpha cristobalite, which is shown to be true for the early Earth. And another question is that if the seawater can allow this high solubility of inorganic phosphorus, can we really dissolve this within this history? We designed this dissolution kinetic model of the phosphorus minerals, and we did a lot of assumption based on the previous work. And basically, it showed that the dissolution of chondritic phosphate minerals could be very, very quick, like within tens of thousands of years. So, which is far shorter than the evolution history of the ocean in the subsurface of the Enceladus. And we are so excited to see that just after 1 year of our publication of our theoretical prediction, Frank Postberg's group actually published the latest evidence from the Cassini data, which basically proved our modeling prediction. It basically showed that within the probably like several hundreds of the acid grains collected by the Cassini, and at least I think a couple of grains has the spectral evidence of the sodium phosphate evidence. So, and based on this, they designed some model to estimate the overall concentration of the orthophosphate, and which agrees well with my prediction. So we will finish the second part of our talk, and this is some summary. Basically, we show that orthophosphate as the dominantly stable species, and reduced phosphorus is not stable, but could be potentially a back signature. And we show that the higher levels of dissolved phosphorus than the modern Earth seawater, which is caused by the high levels of carbonate. but we still don't know a lot of other elements. For example, we know the phosphorus now, but we don't know the sulfur and the transition metals, and also even these divalent metals. We don't know much about this, and our group is currently working on this problem. So I will show you another very key factor for the habitability, which is the availability of organics in the ocean water. So the Cassini, like I said, apart from the inorganics, it also showed a very abundant organic species. And also, if we look carefully into the spectra, it showed that it has a very, a very high variety of the organics, like from C1 to C6, or even higher, maybe to the C7 or even higher. But limited by the detection, like upper limit, we only know like I think the organic carbon less than 150 daltons. We don't know too much about very heavy organic carbons, but for sure there are some. And we were wondering about the possible sources of these organics in the ocean. It could be the leftover from the primordial materials, or it could be brought by the meteorites impact or during the accretion, or it could be synthesized by subsurface water rock interaction, which happens in the Earth's hydrothermal fluid, in the current or the early Earth. So I think all of these three potential sources should face one same question, which is the stability or the synthesis affinity of the organics. Because despite of these sources, you need to know the stability in order to evaluate that this is all a primary signal from which source, or it's actually some artifacts by the detection. So we choose a very simple approach to evaluate this, and we used the chemical affinity for the synthesis reaction. Basically, for the given reaction from the reactant to the product, you can calculate the affinity of the reaction by using this equation, and you can compare the reaction quotient with the K. If the reaction quotient is less than the K, then affinity is higher than 1 than 0, and then the forward reaction is favorable. But if the Q is larger than K, the affinity is less than 0, then the backward reaction is favorable. So we choose some, like based on our previous model, we choose the reactants for the abiotic synthesis reaction, like hydrogen, carbon dioxide, and total nitrogen as the ammonium. And we basically write down the synthesis reaction for these simple organics and calculate their affinity in the instead ocean water. And basically all of these results are based on the assumption that the activity for these organics is 10 to minus 6, which is the detection limit of the Cassini mission. We want to focus on the detectable organics right now. So as you can see, methane has a very positive synthesis reaction given a very large range of the pH. But for all the other, like for several other organics, the methanol is the synthesis affinity is positive in the less alkaline conditions, but become negative in the very alkaline conditions. And with formaldehyde, it's become overall negative, which means that its stability is not favorable. If you form some formaldehyde, it tends to be degraded. The reason why we care so much about formaldehyde is that the origin of life community will think of formaldehyde is the key precursor to form the sugars, as the nuclear base component. Also, with the formate, it has an overall positive affinity given the range of the pH range. we basically calculate all of these hydrocarbons as well as the oxygen-bearing species, and it showed that most of these enzymatic species has positive synthesis affinity, but some special ones like ethane or formaldehyde has the negative synthesis affinity, which means that if you can detect these species, those are short-lived, or even the artifact probably due to the impact of the acid grain. It's very high velocity impact of the acid grain with the payload on the Cassini, which can generate these secondary signals. It basically showed a similar story. And we also conducted some similar calculations on the biomolecules, which are super interesting for the astrobiology community. And the results are not very, very exciting. It's basically show that the amino acid is, for most of the pH conditions, the synthesis affinity is negative. It means that the amino acid tend to be degraded during the water-rock connection in the ocean, which was recently showed by Chuang et al. In Icarus paper. They show that those amino acids are also short-lived. So if someday, probably the next European mission can find some amino acid as the biosignature, that will be very, very interesting. So this is a summary of this part. We basically show that most of the organic species are thermodynamically stable or favorable to form, and the increasing pH will lower will then favor biotic synthesis for all of these investigated organics. And for some key biomolecules or their precursors, such as the HCN and the formaldehyde, they are not thermodynamically stable to form or to persist in Silla's ocean water. So they could be potentially the biosignature in the future missions. So the last part, I want to discuss or kind of summarize about the possible limiting factors for the habitability of the Silla's ocean, which could be also applicable to some other subsurface ocean worlds like Sirius or Triton or those other icy moons. So we already know that the phosphorus, based on my result and Frank's work, phosphorus, the availability is not a limiting factor. It's very abundant. It's much more abundant than the Earth. Based on the Cassini data, we actually detect a lot of nitrogen as ammonium, which is very good for the microbe life to use. So nitrogen availability is not a limiting nutrient. And it is notable that these two elements, like phosphorus and nitrogen, are both limiting nutrients for the aquatic systems on the Earth, but not in the Enceladus ocean. And another one is the building blocks of life, like some key molecules, which, as we see, could be true, because we show here, like the amino acid and ribose phosphate, as well as some precursors for the life, like formaldehyde, and recently we did the hydrogen cyanide, are not stable to persist in the subsurface ocean. So they could be the limiting factor. I want to also go to other aspects, such as the energy for the potential metabolism. And we are also conducting some calculations to evaluate the potential metabolism that can function in the Enceladus ocean. And it showed that the methanogens could be, if microbes can do the methanogenesis metabolism pathway in the Enceladus ocean water, it could gain enough energy for its metabolism. Another one is the sulfate reduction, because the ocean is very reducing. So it's basically we strongly favor the stability of the hydrogen sulfide instead of sulfate. If you have some like active production of the sulfate, and the microbes can use this to reduce it to gain enough energy for them. However, it is also related with another question: is do we have enough oxidants in the subsurface ocean? Because the sulfate is currently on the Earth is delivered by the oxidative continental weathering from the land to the ocean. But like I said, in the insidious ocean, it has no land at all, and the overall ocean is anoxic. So how can you deliver these oxidants into the subsurface ocean, which is a key problem? I think someone is working on this right now, and it is, I think, the frontier of this icy world study is that people argue the cryovolcanism or even the icy tectonics can compact the icy shell, which can deliver the oxidants, including the sulfate, from the surface of this icy shell, and then slowly to the subsurface ocean, which those captured oxidants could be released into the subsurface ocean. But there's no much study available or no strong evidence to support this scenario right now. It's just a hypothesis right now. So I think I'm in a good time. So I will jump into the conclusions. Basically, from this talk, I hopefully persuaded you that the insidious ocean has abundant phosphorus in the dissolved phase and also phosphate phase, which could be used by the potential microbes in the surface ocean there. And the high concentration of the phosphate is mainly facilitated by the high levels of inorganic carbonate, which is similar to the Earth system, like alkaline lakes. And we also show that abiotic synthesis of some organic compounds is favorable, like many of them are favorable, which means that the water rock interaction could be a potential source for these organic compounds. But the properties, the affinity become less favorable at a high pH. And we also show that some key biomolecules or their precursors are not favorable to form, likely constraining the habitability of the insidious ocean. But the future missions could focus on these key biomolecules or precursors as the potential biosignatures. For example, the next generation of the insidious ocean like a mission. You know, my group and as well as many other groups, I think also it's a trend in the European and also American like planetary conference, is that you will see more and more talks about the icy wars, about the ocean wars. So my future work is to constrain the water chemistry and in the end the habitability of those planetary oceans, including the this icy moon or the Mars, the ancient Mars. And the good thing is that a lot of government right now are investing a lot of money into launching the missions to go to this small ocean worlds, such as the European Juicy mission. It was successfully launched last April. So probably it's the 1 year anniversary this month. And this year, NASA will launch the Europa Clipper, which will go, closely observe the Europa. And I think NASA will launch Titan Dragonfly before 2030. It will go to the Titan, and it will it will observe remotely and also maybe land on the Titan surface to analyze the organic compounds, particularly the biomolecules. And China is also planning a Tianwen Force mission to look into the Julian systems, including the Ganymede, Europa, and also Callisto. So we will see. Like, I mean, I'm very happy to see that. I'm very pleased to see that our next generation become more and more interesting in the astrobiology studies, particularly the organizers of this conference. So hopefully, they will mainly participate in these missions and get all the data, and will know a lot of more about this very sick misery icy worlds. So with that, I will stop here, and thank you for your attention, and I'm ready to take any questions.
Thank you very much, Professor, for this was very informative. Now we could take some questions from the comments section. Let me find the first question. Our first question, up until now, what types of compounds were detected on Enceladus plumes, and what information they give us about this moon's geochemistry and potential habitability?
Where can I see the question? Oh, okay.
Can you see it on screen?
It showed here. Yeah. Up to
I think you already answered this question, but
this colleague joined late in my talk. I think I already showed this. This is a very informative study. It's already showed that the Cassini emission detected in the plume that it's basically the water and also carbon dioxide, carbon monoxide, hydrogen, as well as methane, ammonium, nitrogen, and possibly hydrogen sulfide, but not definitely. And it also showed a bunch of these organic compounds, which is still under investigation, I think. And also it showed some salts, which are I think I showed here. Yeah, it showed it's salty and alkaline ice. It's mainly the sodium, potassium, sodium chloride, sodium bicarbonate, and sodium carbonate, and the silica. So given this information, I think it gave a very fruitful evidence about the geochemistry and the potential habitability. Basically, the water will be reducing and alkaline, which is good for the life to form, I think, because one major hypothesis for the original life is that it forms around the hydrothermal vents, especially the alkaline vents, like the Lost City hydrothermal vents. I think the Enceladus water chemistry is very close to the Lost City water chemistry here. Also, we detect a bunch of organic compounds, which are good to form the life. So basically, that's why NASA think and the ESA recently think Enceladus is probably the planet that has the highest possibility to find the alien life there.
Okay, thank you for this answer. Next question. If we started a bacteria or an archaea culture on Enceladus, would it be able to live or even kickstart life over there?
Wow. You mean like migrate some bacteria or archaea there? For sure, they can survive because it's actually I think people are actively working on this topic. They are making some fluid in the lab. Like whenever I went to some conference with a geobiologist, my geobiologist colleague will come to me asking for the recipe for the interstitial seawater. So they will use that seawater to culture the bacteria or archaea there. So I think the even published papers already suggesting that methanogens can well live in the interstellar ocean chemistry. So for sure, I mean, if you can migrate some bacteria or archaea there, they can live very happily. But I think that kind of action is strongly to the space protection policy. So it's not very recommended to do that. So yeah.
But what if we say we ever encounter any life over there by itself? How would it physically look like, in your opinion, given all the material there?
I think we can, the question can be divided into two parts. One is from the original life perspective. Another is from, if life started there, which kind of life can finally be selected. I think both of these two evidence suggest that methanogens probably is most likely microbes there. Because methanogens was thought to be the most ancient, one of the most ancient life on the Earth, and based on the metabolism perspective, also the environment perspective, I think the interstellar ocean water also favors very strongly the methanogens. It has very high contents of carbonate and very high contents of organic carbon and hydrogen, which will favor the methanogens metabolism very strongly.
Great. I will ask another question. What methods scientists used to detect and characterize the presence of organic molecules on Enceladus water plumes?
This is a very good question. So I'm not very good expert on this. Based on my understanding of the Cassini emission data is that it's mainly by the mass spec. It will allow it will collect the acid grain, and the acid grain erupted from the incidus that has a very high velocity. So it will hit the detector very strongly, and it will break up into pieces of organics, which is shown here. It will basically a mass spec. Also, I think I showed here. Yes. No. Yes. Here. Yeah. This is the Cassini data. It will break into parts. Different kinds of the parts of the organics, and based on the simulation experiment my collaborator did in Berlin, they can actually reconstruct the original compounds, likely compounds. So similar like our mass spec detect measurements in the lab, it will break the organics into different smaller species, and based on the distribution as well as the mass to the charge ratio, you can really estimate what's the original recipe for these organics.
Good. I'm going to ask another question. Could presence of certain minerals and salts in Enceladus subsurface ocean indicate the habitability and the presence of potential extant or extinct life forms?
Wow, that's a very hard question. I think this the person who raised up this question is more interested in biosignatures, probably. I think right now for the Earth, like original life community on the Earth, we usually will use the minerals, not the minerals composition. We will see maybe the morphology of the minerals mostly, and also the isotopic fractionation carried by the minerals. to see whether it was life left over or not. Because inorganically, abiotically, the water rock interaction can also generate a lot of minerals, can also alter these things. I think one potentially important, promising mineral to look at is pyrite. So for the water interaction products we saw in the comets, we usually will see pyrrhotite, which has a very different composition as well as isotopic signature with the microbially generated pyrite, because the pyrite can carry the sulfur isotope composition, along with the microbial activities. So I think one could potentially look into the pyrite. Others, I don't think there are any good minerals for this perspective.
Okay. Let's move on to our next question. Can insights to Enceladus' origin, evolution, and potential habitability be determined by its isotopic analysis, and what isotopic ratios would be of interest for the researchers, or of interest?
Yeah, this is a very, very good question. So I think if ESA can really launch the lander on the Enceladus, they will put I would imagine that they will mainly look for either biomolecules there, and also measure the in situ the isotopic fractionations there. So depending on the different scientific goals, you can focus on different isotopes. For example, if you focus on the water evolution history, because Enceladus is continuing losing the water to the space. If you want to look into that history, want to reconstruct that history, you can look into the hydrogen to deuterium ratio. And if you want to look into the outgassing history, you can look into the noble gas isotopic ratio, like argon or the noble gas isotopes. If you want to look into the biosignatures, it's strongly to look into the carbon ratio, because as everyone know, life will prefer to use lighter carbon, and will cause the carbon isotopic fractionation. So if you want to look into for example, element cycle, of nutrients, you may look into the nitrogen isotopic ratio, also sulfur isotopic ratio. So I mean, I think by then it will heavily depend on which kind of the isotopes you can measure. I think all the isotopic ratios are very important and will show very surprising results for sure.
Great. Our next question is, how can we predict the presence of water and water cycle on a planet or a satellite? What data are needed, and to what extent? Which telescopes are actively researching of this question? Which of these telescopes share their data as open source?
It's a good question. So I think for our
Professor,
solar
can
system,
you hear us?
I can hear you. Can you hear me?
Okay.
Yeah. So for our solar system, is very well understood. The water cycle. So if you are familiar with the origin of our solar system or the evolution, basically outside of the snow line is full of water. It's very rich in water. So basically rich in hydrogen. So the second question is about the water cycle. The water cycle is composed of two parts. One is the outgassing to the space. You can easily detect that using some space physics method. Another one is whether it has liquid water down there. I think it's still an active field, because even this year, I think people are still arguing that some very small icy moons may have active liquid water below the icy shell. So what data are needed to constrain that? I think you need to know firstly the radius of the planet. Like I said, for the solar system, we already know that beyond the snow line, it has a lot of water. But I think it's an active field to look into the exoplanets right now, because the exoplanets are so different. They have so different kinds of the exoplanet systems. So looking for the water is the key target right now for the exoplanet mission. So you need to constrain first the mass, also the radius, and you also need to know the distance between the planet to its star, like the distance between Earth to the Sun, right? You need to know the distance between that exoplanet to the star, because that can tell you whether the planet can hold the water in the planet, as well as can support the liquid water, can have a warm climate to have the liquid water on the surface. And I think we have a lot of data already from the several space telescopes launched by NASA. The retired ones are Kepler telescope and the Hubble telescope. And right now, the James Webb telescope is really aiming to that field. Is that it has a higher resolution to show this water signal and to really look into the potentially habitable and with liquid water on its surface, the planets. So I think you can, if you can have the resources, probably you can propose to the James Webb team to get some data and analyze them. That would be very, very helpful.
Okay. Next question is going to be, is there a sign of a large-scale asteroid impact on Enceladus that could lead to the extinction of possible life forms in the ocean before, or any other possible cause of mass extinction?
That's a very good question. Yeah, that's a very good question, especially the second part. So the first part is that it's not like Earth. Like I said, it's covered by the thick layer of ice. So I think the asteroid impact may induce the melting of the ice regionally, but it won't cause. It couldn't cause any mass extinctions. Also, I think a mass extinction is a term mainly for the plants as well as the animals. The microbes, they can survive very harsh environment. So I don't think there is any extinctions in the Enceladus ocean caused by the impact. Not like dinosaurs, because dinosaurs are so fragile. And if there's any possible cause of mass extinction, it could be the very strong disturbance of the Saturn system. For example, if there are some very strong disturbance of the Saturn system, it will change the tidal force to support the liquid water in the Enceladus ocean, which may cause it totally freezing. If there's any life there, it will be frozen. It will be gone. So, but I don't think that's a very large possibility because Saturn has so large mass. It's not very easily to disturb.
Great. Our next question is, what is the level of contribution of Enceladus hydrothermal vents on its water chemistry and habitability?
That's a very good question. Basically, we don't have so much evidence for that. Only recently, people proposed some geophysical modeling on that. I think it's a group in France who did the modeling, showing that the hydrothermal activity, it can alter the whole ocean, probably in the time scale. I don't remember correctly the number, maybe wrong, but please go and check the paper. Maybe in the order of 10 to 5th to 10 to 7th years. So the hydrothermal activity will alter the whole ocean. So basically, the hydrothermal activity could significantly impact the global ocean water chemistry in the ocean. Yeah.
Our next question, and before that, Remzi is thanking you for the amazing work, and he's saying absence of the land makes me think about the surrounding moons. Is it too superficial to think of a way to collide them using nukes, etc., nuclear power, in case that Enceladus is inhabitable?
I'm not fully understand the question. Like, are you talking about
could we use nuclear power? Nuclear weapons to collide those moons into each other? And would it be beneficial to start the life? Perhaps, I don't know, because it's frozen. I guess.
Are you Elon Musk? I mean, yeah. I mean, people like Elon once proposed that use nuclear weapon to hit the Mars to make it more habitable. But I think if you can push the icy moon to some region, it could become habitable. Like, for example, the Callisto right now, people will think it could have liquid water there, but it must be very limited. Or another group of people will think it's totally frozen. So if you can push the Callisto towards the Jupiter, then it may have the higher tidal force, stronger tidal force. Then it may support the active water rock connection down there, even the hydrothermal activity. So it may become habitable. So I think it's a crazy idea, but it's a feasible idea. But
like on Mars.
Yeah.
Okay, and I think it's our last question. I would like to ask the professor about tardigrade research on astrobiology. Even though there has been few space missions, some questions remain unanswered. What is he thinking about tardigrades? Maybe this question was about, would tardigrades be able to survive on Enceladus?
Tardigrades, okay. It's actually, like I said, it's highly unlikely to involve any animals there because it's too harsh for animals. It has no land, and it's very alkaline condition. I think not only about the animals, it's also very hard to evolve even cell there because it's full of water. So how can you form these big polymers in those water world? That's a very big challenge. So not alone the animals. So I think like you said, the tardigrades may form in some planetary system which has lands and ocean, and big lands and big ocean, like Mars, ancient Mars. But I don't think it's likely to evolve in the icy moon. But you may keep looking for the exoplanet, because exoplanet has so many exoplanets out there, and many of them are potentially habitable. So maybe origin of life and evolution of life is not only happening on our Earth, may also happening in some other exoplanets. So
okay, I hope Professor answered your questions, and if we don't have any other questions, we thank you for joining us today, for sparing us
Thank you.
your time from your busy schedule, and we hope we will see you next year and the next year and the next year with your new researches.
Thank you for
And with
organizing
that,
this.
thank you for it too. I'm withdrawing you from
Okay.
the screen.
Bye-bye.
So you can remain to watch. Thank you and bye.
Bye.
We will thank you all for listening to our first session. We will be back with Dr. Michael. He will be talking about extremophiles on Mars. And we'll see you soon in 5.
Hi everyone, welcome to the second session of our conference. I'm Suda Kalın and I am a fourth year biology student in Marmara University. Today I will be hosting our second session. Our second session will be with dear Dr. Michael Macey. Let's get to know him before our session. He's a microbiologist and a research fellow in the Astrobiology Open University group. His research is currently involving investigating the diversity and activity of microbial communities, inhabiting extreme environments that are analogous to Mars and other extraterrestrial sites. He also performs simulation experiments to assess the growth dynamics and survival of microbes in simulated extraterrestrial conditions. Currently, he is a research fellow, lecturer, and postdoc at the Open University in England. Now, I would like to bring him to do his presentation on the possible life of extremophiles on Mars. Hi, welcome.
Thank you very much.
How are you doing?
I'm very well, thank you. I am setting up to be able to share my screen.
Do you need any help with that?
So I've got share entire screen, but it's not letting me. The share button itself is grayed out.
Okay, so you can either press present and slide.
I'm so sorry. I now realize what I was doing. Yes. Okay. I now realize what it was I was doing.
Sometimes you need to allow also some securities from your Chrome tab or Internet tab. Yeah. For that, you need to allow your share. Okay.
Yeah. Can I confirm if my screen is coming through?
Right now, no. It's not. Yeah, it has come. Okay.
Okay, fantastic. Okay, apologies for the slight technical delay there. But okay, let me go back to the presentation, get away from inception of the slides. Fantastic. Well, thank you very much to... Sorry?
No, it looks great. The slides are looking great. Okay. It's okay.
Brilliant. Brilliant. Thank you very much. And yeah, thank you to the organizers for allowing me to present some of my research as part of this really exciting event. It's always so interesting to see how much astrobiology is growing as a field. I think it's a field that really resonates with people because, you know, is there life potentially outside of Earth is probably one of humanity's oldest questions, and it's one that we're still wrestling with. So this is a recent image of the surface of Mars sent back by the Perseverance rover, showing the surface of Mars today, what we know to be an inhospitable environment. It's incredibly arid, it's cold, there's next to no atmosphere, and it's bombarded by high levels of both ionizing and ultraviolet radiation, which along with a few issues of chemistry make the planet, you know, incredibly hostile. But we know that from the last few decades of Mars exploration that this was not always the case. There are actually extensive bodies of evidence that Mars was once a water-rich planet. There's been minerals detected that only form if there were once the presence of water. And from this, we can try and extrapolate where these waters would have been spread across the planet's surface. So this is just one potential projection of where there would have been different lakes and oceans on the surface of Mars. And it's an incredibly exciting time for Mars exploration. So in 2020, we saw the launch and landing of both the Perseverance and the Jurong rovers. The Jurong rover is now inactive, but it was able to gain extensive evidence supporting the existence of these former Martian oceans. And along with Curiosity, the Mars alumnus running since 2012, Curiosity and Perseverance are still generating extensive data sets from these formerly water-rich environments. And it also means that in 2020, there were 3 rovers active on the surface of Mars. As we look to the future, in 2028, we're expecting the launch of the ExoMars Rosalind Franklin rover and the full analytical suite that will be taking to the surface of Mars. And in the 2030s, there's also proposed Mars sample return, the collection and return of Perseverance's sample cache, representing the first ever non-meteoritic materials to be from the Martian surface returned to Earth. So it's an incredibly exciting time for Mars exploration. So the makeup of this talk is going to be a refresher for some key microbiology concepts. For those of you with a biological background, it's just going to be a quick recap. And for those of you who have less biology training, it's going to be a brief introduction into certain key concepts within microbiology and our understanding of extremophiles, why living in extreme environments is necessarily stressful, and how to respond to those stresses. We'll then be going on to research that I perform, as this was mentioned in the introduction, on analog environments and simulation experiments. So how do we study Mars from the Earth? So key microbiology concepts. We have an amazing diversity of elements that we know about within the universe, but if we look at what is common between all living things, between all cells, the diversity is significantly reduced. Chinops, so carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur are found universally in all living things and also make up the majority of all cells. Now, that's not to say that the other elements aren't important. There are many enzymes and proteins that require specific metals to function, but it's these 6 elements that are most important to life as we know it. Now, we also know from looking at modern life and extrapolating backwards, our understanding of life shows that it requires 3 key components. So you need a system to enclose molecules. You need to have an internal environment that's separate from the external environment. You need an ability to transfer information between generations. And you also need a means of generating energy effectively to sustain the 2 previous. And we have membranes that in bacteria are encoded or provided by phospholipids. So we have, let me get the laser pointer up, pointer options, laser pointer. We have phospholipids. So we have the phosphate head, which has a negative charge here that makes it hydrophilic. It can interact with fluids. And then we have the hydrophobic tail in the form of the saturated fatty acid and the unsaturated fatty acid, which has the kick. Now, if you take a pot of phospholipids and you dunk this into water, you would see the formation of these spherical structures because the heads can interact with the fluid and the tails cannot. So you get these single layer lipid spheres, or more interestingly, these bilayer spheres. So in the absence of biology, you now have an internal environment that's separated from the external environment, and this will have fluid captured inside. So it's this lipid bilayer that makes up the membrane or variants therein that provide the separation of an internal chemical environment from an external, harsher, less maintained chemical environment. Or think of it simply as a microbe floating along in the ocean. Its internal contents need to be separated from the external contents. Combination of these 2 is effectively the microbe is dead. System of information transfer. All life on Earth uses DNA. So here we have a schematic of a nucleotide. So again, we have phosphates and sugar. So phosphate being charged here. We have the sugar, the deoxyribose, so the D in the DNA, and then here we have the nitrogenous base. So in living organisms on Earth, we have 4 nucleotide bases that are used. So adenine and thymine, which will form 2 hydrogen bonds with each other, which is why you get this pairing shown here of orange to red, and guanine and cytosine, where you get 3 hydrogen bonds between them, between opposite strands. There is some really exciting work that's being done in America that's funded by the Templeton Foundation, and they're looking into could alternate nucleotides work? Because whilst these are the nucleotides we observe in all life, and with RNA we see uracil instead of thymine, that's a very minor difference between the 2. Other nucleotides exist, but they aren't used by life, and there's interesting theories as to why. And then we have systems of generating energy. So this is a schematic of an amino acid. So we have an amine group on one side in green, and we have the carboxyl group on the other in yellow, and we have R representing the variable side chain. And again, there's a massive diversity of amino acids, but all life as we know it uses 20, 21, depending on if it's a specific set of experiments or not. Now, amino acids do this really interesting thing where when you have 2 amino acids come into contact, the amine group of one interacts with the carboxyl group of another. This forms a peptide bond. And this will continue happening. You get longer and longer peptide chains. The amino acids keep bonding. And because of the interactions of the side chains, you start to get folding interactions and more complex structures until you eventually end up with more complex structures as shown here. So you have the repeating loops, you have the bends, and this is the structure of an enzyme responsible for cycling of sugars inside of cells. But it's enzymes in modern cells that perform the processes for generating energy, maintaining the internal cellular environment, so pH, as well as allowing the transfer of materials across the membrane. So once you have these three components, then you get to a self-sustaining chemical system capable of Darwinian evolution. So Darwinian evolution owing to the DNA and its transfer between generations, self-sustaining because of the actions of the proteins inside the cell, but also because of the lipids maintaining an internal chemistry separate from the outside. It's also the self-sustaining part is why we're not considering viruses as living under this NASA definition of life, because they require a cell in order to replicate. If you remove their host, they can't replicate. They're not therefore self-sustaining. There's a lot of discussion around whether this is too limiting of a definition, but definition of life itself is an incredibly complex thing. So not all life is the same. Some organisms are able to inhabit environments that others are not, and those extreme organisms are termed extremophiles. And it's always fun to turn to this extract from the Martian Chronicles, written by Ray Bradbury, when we consider what an extremophile is. It's a discussion between a husband and wife, Martians living on Mars, discussing if there could ever be people living on the third planet. And the husband responds that the third planet's incapable of supporting life because our scientists have said there's far too much oxygen in the atmosphere. Because, of course, extreme is a relative term. We consider extremophiles as extreme loving organisms, but the extreme is relative to us. So for things living in a hot spring, if they were capable of greater thought, they'd probably consider us living in a room outside of a constant body of liquids at room temperature as extreme, but we're the ones coming up with the definitions, and so this is the starting point from which we base all of these. So it's a physical or a chemical extreme. So a physical extreme would be too hot or too cold, or a chemical extreme would be something like salt or pH. And most extremophiles, and the most extreme of the extremophiles, are single-celled organisms. So we're interested in studying these types of environment and the organisms that live within them because it's genuinely the study of life at its very limits. And when we look at the organisms within those environments, they often possess adaptations to these extreme selection pressures that result in genetic traits, in proteins that aren't found elsewhere on the tree of life. So it's an amazing way to study evolutionary processes. And to that end, it's also very important to our ability to understand habitability. So there's this fantastic figure from Marino et al., which shows against just 4 different environmental parameters, so salinity, temperature, pH, and pressure, all known life. So in dark green, we have everything that's been isolated into pure culture that we know about and can examine in a laboratory and really poke with a stick to see what exactly it's doing. And then in the lighter green, we have environmental-based studies. So things that we know about through sequencing-based studies and the analysis of DNA from environments where we observe life, but we haven't necessarily been able to isolate organisms. And we can see that we have life at these very edges of these boundaries. But then when we look at all environments on Earth, there are, of course, environments that exceed these boundaries. There are pressures that are too great. There are temperatures that are too high and are in fact used for sterilization processes when we have to have equipment that is clean. So working in a laboratory or working in a hospital. And it's also very interesting if we then take this concept of habitable environments across former and current Martian environments, and we see that there is, in fact, potentially overlap. Not saying that there is life on Mars, but saying that there are potentially or could have been habitable environments on Mars. And then lastly, possessing evolutionarily unique adaptations also means that some of these microbes will potentially possess unique antimicrobial compounds. And so this is a major area of research, both within our group and within the broader research sphere, in trying to identify and isolate novel antimicrobial compounds in the fight against antimicrobial resistance. So if we go back to our hot spring, The organisms that live here are termed thermophiles, so thermo meaning heat and phile meaning love, so thermophiles. You're a thermophile if you survive above 45 degrees centigrade, and you're a hyperthermophile if it's above 85 degrees centigrade. There's often this gradation, even between extremophiles. And the problem that they encounter is one of physics. So because of the great levels of heat, the great levels of thermal energy, the membrane can simply vibrate apart. Now, if we think about what life requires, you need a membrane, you need maintenance of the internal environment by proteins, and you need the ability to transmit your genetic information to the next generation. So the membrane vibrating apart, the cell is dead. If the proteins denature, which means they lose their structure, they lose their function, the internal environment cannot be maintained, and again, you're dead. And DNA can also be damaged as a result of the energy. And if we consider most mutations that would happen randomly will be neutral, they will occur in either non-coding regions or not have significant effect, but they could also be deleterious, much more likely than they would be beneficial. So how do microbes respond to this? Well, in addition to the phospholipids that make up the membrane, you can have alternate fatty acids and other molecules. So if you add isobranch chain fatty acids, it makes the membrane more solid, better able to resist the impact of the thermal energy. The proteins are better maintained through the synthesis of heat shock proteins, proteins that bind to other proteins and help them to maintain their structure and their function, and DNA repair proteins. So there's no way to prevent the DNA actually getting damaged as a consequence of the high heat, but there is a way to ensure that you can repair it. And this is very similar to what microbes do to radiation damage. It's not that they're resistant, it's that they're able to repair effectively afterwards. Now let's go to the other end of the spectrum. So psychrophile, cold-loving organisms. This is color peak. This will be reoccurring throughout the talk, and this is an incredibly cold environment on an island in the Canadian high Arctic. You're a psychrophile if you grow at temperatures below 10 degrees centigrade, and here the problem is effectively the opposite, as you would expect, to that approached by the thermophile. So the membrane can freeze, and if it freezes, then of course it can break and it will kill the cell. You can also get salt crystals forming inside the cell that will rupture the membrane. And you also have the fact that in the same way proteins can vibrate and denature and falling apart, they can also just lose activity. They can slow down as a consequence of these low temperatures, and then the internal chemistry is not being maintained. And there are similarities in the adaptation. So in the same way that the thermophiles change the makeup of their phospholipid membranes, you also see that with psychrophiles. So you have short chain and unsaturated fatty acids being incorporated into the membrane, and they actually make the membrane more fluid and better able to resist the impacts of these low temperatures. In the same way that when we go hiking, you're told to take something sugary with you, and they sell camping snacks that are rich in sugar, microbes will build up sugars to prevent freezing. It's this release and breakdown of the sugars helping enhance the temperature of the cell. You also get cold shock proteins, which bind to proteins in the same way heat shock proteins do, but to help maintain continued function. And you also get antifreeze proteins. So these bind to tiny salt crystals when they're in the very early formation stages and prevent them from growing. Now, if we then go to a chemical extreme, we have the halophiles, which are the main area of my research, and these are salt-adapted organisms. So hypersaline environment in which these organisms occupy, you're hypersaline if your salinity exceeds that of seawater. But we can see that there are in fact environments that greatly exceed those salt concentrations. So shown here is a map of the different countries in which we work with universities within, and where we analyze some of the hypersaline environments, getting an understanding of these halophilic organisms. So there are salt flats in Spain. There are salt flats in the Sahara Desert, also in northwestern India, in Kutch. There are salt lakes in Ethiopia, and there are salt pans that are the remnants of a megapaleo lake. So they are globally distributed, and they have salt concentrations greatly exceeding that of seawater. So what is happening to the cells within these environments that's so stressful? And again, it's a physical process that is killing non-adapted organisms. So if you have a cell in, let's say, a less extreme environment, red representing salt here, water can move across a membrane, and water will move across the membrane to the greater concentration of salt. So in a normal environment or a non-hypersaline environment, you'll have movement across both ways. In a hypersaline environment, there's a lot more salt molecules on the outside, so water will move across the membrane in much greater amounts than it will move back into the cell, and you will lose your internal chemistry and the cell will die. Now, there are two different strategies that you can take here, salt out or salt in. And fascinatingly, we see that this varies depending on whether you're bacterial or archaeal. In bacteria, they try to keep the salts out. So to prevent this movement of water, they build up organic compounds known as osmolytes. And this balances the internal chemistry of the cell without having masses of salt imported. What archaea do, or the haloarchaea specifically, is they allow the salts in. They just allow the salts in. That solves the issue of the water going both ways, and they just simply have proteins that are able to continue functioning in these high salt concentrations. And then there are, of course, the polyextremophiles. So if we go back to Colour Peak, our example of a cold environment, well, it's not just cold. The air temperature when this photo was taken was below 0 degrees, but we can see that there are small pools of water present in this spring, and they're present as liquid water because they have high levels of salt in them. So any organism living in this environment is adapted to both the impact of the salt, maintaining the internal chemistry in the face of this, but also to the cold, making sure that they maintain their structure of their membrane and activity within the cell with these cold shock proteins, the sugars, and the antifreeze proteins. And the additional machinery that requires is an example of the unique adaptations possessed by extremophiles, but also about how extremophiles have quite an energetically expensive lifestyle, because you have to be constantly maintaining these structures and these processes and these compounds to not be overcome by the environmental selection pressures. And this brings us neatly onto analog environments. So are all extreme environments analog environments? Well, it depends on perspective, because an analog site is a region possessing a chemistry approximating that of a celestial body of interest. So that's a lot of words, but let's break them down. So approximating. Let's say we want an analog environment for Mars or for one of the icy moons. Why would we use the term approximating? Well, there are two key reasons. One, we do not have absolute certainty of these environments. Even on Mars, such a well-explored planet, we're still working out from chemistry, and we're making inferences based on mineralogy what this environment exactly is like. We do not have 100% certainty, the same way I do, of going out to my garden and taking as many measurements as I want. We don't have that luxury. We have entire scientific suites of equipment on the surfaces of some of these bodies, but we don't necessarily know everything about them. So we work with the information we have, and also approximating, because even though we have this incredible wealth of fascinating natural laboratories across the surface of the Earth, we're an oxygen-rich planet. And even anaerobic sediments on Earth are impacted by this oxygen, because there'll be microbes in the surrounding areas that will produce nutrients that will enter those sites, as well as minerals that will potentially end up being subducted into these anaerobic sediments. So oxygen is always a relevant factor when we consider Earth. So we're always working with chemical proxies instead of something very chemically accurate. We're working with what we have in order to gain a greater understanding of both the inner and outer spheres. So the surface of Mars today, we found bodies of salt on the surface. We know it's incredibly oxidizing. Anaerobic, we know that there's hypobaric pressures, so incredibly low atmospheric pressures, and it's bombarded by not just UV, but also ionizing radiation. And on top of that, there's a brilliant paper by Wadsworth and Kekel, where they show that UV radiation and the perchlorates, the chemicals that are abundant on the surface of Mars, together are incredibly bactericidal. They can destroy bacillus spores, which are often used as the benchmark for extreme survival. It's also relevant to consider that an analog environment is an analog for a celestial body at a point in its history. So this would be an analog for the surface of Mars today, but as we've already discussed, Mars in the past would have been a much more water-rich planet. And what's also fascinating about this is Mars would have had these waters in what on Mars would have been the Noachian era, which overlaps with on Earth the Hadean and the Archean era. So the theory is that life originated on Earth predominantly in the Archean era, because the Hadean era, as the name would suggest, including the term Hades, was on fire. It was balls of magma at that specific time. So it's a really interesting variation, because it's effectively the inverse now, where we consider Mars to be completely inhospitable. There would have been some oxygen in the atmosphere of early Mars. This is a source of great debate whether it actually would have been more abundant relative to other components, or whether simply having an actual atmospheric pressure would mean that there is more oxygen because there's more atmosphere. But having an atmosphere would also mean shielding from radiation. So we also predict that these waters would have been still salty, and following the loss of the atmospheric pressure, they would have been increasingly salty, moving into the next era on Mars known as the Hesperian. So Axel Heiberg Island and the springs therein are considered analogues for Mars. So it's located in the Canadian High Arctic, and it's in the region of continuous permafrost. So the white coloration here is the permafrost that's constantly on the surface, and it's known as a polar desert. So it's very cold and it's very dry, with the exception of these springs and the upwelling in them. So Colour Peak is an environment that I've worked on more extensively with the University of St Andrews, and also collaborators at the Open University. But there's extensive work that's also been done on Gypsum Hill, Wolf Springs, and Lost Hammer. So some of these other springs marked by the red circles. So the springs are both hypersaline and sulfur rich, two components that make them good potential candidate analog environments. They're also sulfide rich. If we look at the black coloration that's present in the mud here, or in the sediments more accurately, this is sulfide. So if you've ever smelt rotten egg or walked past a blocked drain, that smell of sulfur is actually specifically sulfide. So Colour Peak has previously had some studies on it referenced here, and we focused on this environment because advances in microbiology hadn't specifically been applied to this environment. So we were able to take advantage of advances in sequencing platforms to get a greater level of characterization of the microbes that lived here. And by studying here, trying to gain a better understanding of potential habitability and maybe viability of metabolisms on former Martian waters. So the first question we have to ask is it relevant? So ignoring the graph for now, it's always worth starting with the physical and the chemical parameters of an environment. So the temperatures of Colour Peak fall within the temperature range for colder temperatures predicted on early Mars. So there are effectively two models with variation therein, that Mars used to be warm and wet, so 20 degrees centigrade with water on its surface, or cold and wet, so experienced occasional freezing temperatures but still had water. So this fits within the range for one of these models, and this is what I was talking about in terms of approximating. We're working with the information that we have and with our best models. Now, if we look to the graph shown here plotted, we have the three most abundant elements in the Colour Peak waters, and these are represented by these red dots here, both our study and this black dot from a previous study. And you can see they cluster very much down here. That's because sodium and sulfur are the two most abundant elements, and then there's quite a lot of calcium as well, but nowhere near as much as the other two. If we focus on this here, the other grey dots that are present on this figure represents other terrestrial analog environments, such as the Deccan traps in India. So we see those up here. So these are other hypersaline fluids. We also have a range of modeled chemical fluids. Now, two I want to draw your attention to are these diamond shapes. So there's one diamond here and there's one diamond here. Now, these were modeled by one of my colleagues and one of the co-directorate of my group that I'm in, Astrobiology OU, Dr. Susanna Schwenzer. So Gale Crater is a believed former lake on Earth, and there are evaporitic minerals that would have been formed as a consequence of the former presence of this water. Now, using modeling in geochemistry, we can extrapolate backwards. Okay, what would the chemistry of this water have once been like? And we see that they cluster quite closely to the chemistry of the Colour Peak water and sediment chemistries. So they're very close in terms of the most abundant elements to these predicted Martian water chemistries where the Curiosity rover has done a lot of analysis. So we consider, again, with an approximation, the broad strokes, this is an appropriate physicochemical proxy for early Mars. So the question is then, what microbes are surviving in this environment? In order to analyze this, we extracted the DNA from the microbes in the sediment and we sequenced a conserved gene that's present in all bacteria and all archaea. We did this from replicate sediment samples, sent this off for sequencing, and the main thing here is not to spend too much time focusing on the various taxonomic categories and the different Latin names. The most important thing is looking at this graph, observing the colors, and seeing that purple bars dominate the graph. Why is that interesting? What do they represent? Well, purple bars represent sulfur oxidizing bacteria, and their high abundance. You've got a lot of this specific genus of microbe, but you also have the other pink and purple bars present on the graph. These all represent sulfur oxidizers. Now let's take a step through what they are. These are microbes that get energy by the oxidation of reduced sulfur compounds. And this is something that can be done in the presence of oxygen, as we do through aerobic respiration. So we use oxygen as our final electron acceptor, or using nitrates in an anaerobic way. Now, this is cool for two specific reasons. Sulfide, in addition to sulfate and sulfur, has been detected on the surface of Mars. So sulfate is incredibly abundant, as is sulfur. Sulfide has been detected and is present in Martian meteorites that have reached Earth. Nitrate is also present on the surface of Mars in reasonably high abundance. It's been detected in the mudstones by the Curiosity rover. Now, these organisms, the most abundant of them that we've detected within this community, are also what's known as autotrophs. So if we think of what a plant is doing in order to gain energy, it's an autotroph. It's converting carbon dioxide to organic carbon. And these microbes are doing the exact same thing. They're not heterotrophic like we are and some of the other microbes we detected. So they require organic carbon as we do in order to be able to grow. The problem we have with studying environments like this, however, is being cold and being very salty. Those are two properties that are very good at preserving DNA. So it's possible some of the microbes died millennia ago and their DNA has simply been preserved and we're detecting them. So what we can do instead is look at RNA, and by looking at RNA, something that is much more easily broken down in the environment, it has a very short half-life. Even in cold environments, we'd still consider it to be on the maximum of days. So if we do the exact same analysis, we can see that, yes, the diversity has pretty much collapsed. So we were detecting some dead microbes, but the active microbes are mostly sulfur oxidizers. So this is a nice result. This confirms that we're detecting, reasonably a representative community. And it's the same few key players that we're detecting here. So the same sulfur oxidizers, the same autotrophs, with some other diversity hanging on. Now, I work predominantly with geologists and people who don't enjoy a lot of microbiology. So this is a constant question that comes to mind: is why is this interesting? Why are we interested that this group of microbes grows in this specific pool of water in the Canadian Arctic? And why are we even interested in studying analog environments? And it's because they're useful to generate hypotheses. Now, if we go further into the ecology of this, let's look at why this result is interesting. This is a picture of an arch with the keystone in red. So this is the central part of the arch and is the thing that keeps it up. Now, this is a concept in ecology as well, where you will have some species that are maintaining the entire ecosystem. In a patch of grass, and this is the small patch of grass I worked on for the 4 years of my PhD, the grass itself would be considered the keystone species. Not always the most abundant, but it's one of the only organisms present within the system. It's driving most of the photosynthesis, most of the input of carbon into the soil, so we could reasonably consider grass as the keystone species here. If we go to our Arctic spring, it's this same purple bar. The sulfur oxidizers, specifically the autotrophs, that we could maybe consider the keystone species. So let's put everything we know about this environment into context. We know that these microbes are growing on the sulfide, the black coloration that's present in the sediment, and we know that they're fixing carbon dioxide as their source of carbon. So that's not too much extrapolation so far. Now, as these microbes are growing, they'll be releasing carbon to modify their environment, either intentionally or accidentally, because some cells simply leak certain compounds. If we think of formaldehyde, that is something that cells can quite readily lose. Also, as they're growing, they'll be dying, and this represents a pool of organic carbon they themselves cannot use. But we know that other microbes are present within the ecosystem that actually do need organic carbon. So this would potentially represent a pool of additional carbon, either through necrophagy, one of my favorite terms, feeding on the dead, or feeding on secreted organic carbon. Now, we know that these sulfur oxidizers are growing on carbon dioxide and using either oxygen or nitrate as an electron acceptor for the completion of their sulfur oxidation. And what's very exciting is knowing the chemistry of the modeled Gale crater waters and knowing the atmospheric composition of Mars now, so the concentration of CO2, the concentration of oxygen, the level of nitrates detected in the mudstone, we can use something known as Gibbs free energy calculations to work out, okay, just considering how much carbon is required, how much sulfur, the oxygen or the nitrates, would this metabolic reaction be thermodynamically viable? Would any metabolic reaction be thermodynamically viable under this proposed former Martian chemical environment? And the answer is yes. Sulfur oxidation is feasible under these modeled conditions for early Mars, and it was in fact one of the most viable of the metabolic reactions in an anaerobic context. So we have potential keystone species not only in the ecosystem, but we can extrapolate that were these chemistries of our models accurate for the former Martian waters, perhaps something like an autotrophic sulfur oxidizing bacterium or something that utilized these metabolic pathways would be viable under these former environmental regimes. Now, we then can take this one step further if we consider testing this viability. But how do we test if these microbes can grow in Martian chemistries? And that's through simulation experiments. So when we simulate, we're performing simulations to try and get a better understanding of the boundaries of habitability by effectively making Mars in a bottle. We're testing the physical and chemical conditions under more accurate regimes than the chemical proxy that the analog environment gives us. And it also gives us the ability to try and look for biosignatures, evidences of life that could occur because we have microbes growing. And specifically for the context of the experiments that we performed here, by simulating a Martian chemistry, I mean we're simulating two different geologies. Mars is a planet, and we wouldn't consider the geology of Earth uniform. We have to take that same approach when we study Mars. So we have a representative collection of minerals that gives us within 5% chemical accuracy of the chemistry of Gale crater, this curiosity analysis site that's considered representative of a broad global Martian chemistry. And then we also have a more iron-enriched chemistry, that red color that we associate with the surface of Mars. So this is work that was done by my former student, James Oliver, as part of a funded summer studentship. So what we were doing was testing the question of, can color peak microbes grow under Martian conditions? No, this is an analog environment we're arguing. So when we take those microbes and we put them under more accurate Martian conditions, they should grow. And our sulfur oxidizers is growing. Two very simple questions, but important questions built from the hypotheses we've generated. Now, if we look at the microscopy results that we generated, we can see that it took a while, but there was growth. The circles go up, they're higher than they were at the start. So yes, is the very simple answer. They were growing. Fantastic. It's not a shocking conclusion because the waters of early Mars were undoubtedly would have been habitable. The question of whether life was ever there is a completely different question. Now we then have our color peak microbial community, the color peak RNA community. So if we look at what those cells that have grown are, what do we see? Well, we see that the answer is yes, the sulfur oxidizers are growing. So this is the rock nest chemistry here. We see that purple is still present in the community. We have the other hangers on. So these are heterotrophs, just other anaerobic microbes that are growing under these simulated rock nest Gale crater conditions where the Curiosity rover is analyzed. So it's growing. Okay, that's a fantastic result. This metabolism is technically viable under Martian chemistries when we're testing it, when we're taking it outside of the model. There is an asterisk on this for a very specific reason, and that is it doesn't happen in fact, most of the simulations we ran. And we instead see that the purple is pushed out, and in most cases pushed out by these red bars. So the yellow and the orange and the blue are just other heterotrophic organisms, not doing anything specific. But in the red is also present here, the red, and the red. These are microbes that are known as sulfate reducers. So this came up in the previous talk. They do the reverse reaction. So these are the things that are making the smell in the blocked drain. They convert sulfate to sulfide. Now, this is an interesting result because this here, the purple still being present after the simulation experiment, shows that yes, sulfur oxidizers are viable. But because we've taken this community approach, we've added this milieu, this mixture of cells from an environmental inoculum. We've added in a lot of different variables here. So we've also shown that a lot of heterotrophic fermentative bacteria would be viable under this chemical regime, but also the sulfate reducers, which is something that our modeling from the previous paper also showed sulfate reducers would have been viable. And the question is then, is it that the sulfur oxidizers simply cannot grow in this other, more iron-enriched chemistry? Is it that it's more selective for sulfate reducers? Or is selection not a factor here? Is it simply that if we ran 100 variables, sulfur oxidizers would have grown in 10 of them or 3 of them? And it's just a simple consequence of providing a regime that some microbes can grow better than others, but also accounting for the potential role for neutrality, for stochastic chaotic variation. This is published with James as the lead author in the journal Life. And it was an interesting follow-up study to the Color Peak work because it's challenging our hypotheses, giving us partially the result we were expecting, but also very much the result that we want. So to wrap up, the key point at the start is that all modern life requires a membrane, a way of transferring information between generations, and a way of generating energy. Microbes can survive extreme environments, and by studying them, we can learn about the habitability of other planets, with these analog environments representing these natural laboratories that give us this insight. We can also do modeling and simulation experiments to further the insight provided to us from these analog environments and challenge these hypotheses about what could have been viable elsewhere in the solar system. So yeah, thank you all for paying attention, and I'm very happy to take questions. And I will also try and get back to the screen I was on and stop sharing my screen. There we go.
Sorry, I was muted.
That's all good. That's all good.
Well, thank you for this very informing presentation. It was so nice to listen and learn more about astrobiology. And if you're ready for the questions, I can take some of them.
Absolutely.
Okay, we have actually quite a bit. And the first one is, what kind of detailed research should be done to understand what types of microorganisms could live in the deep underground oceans of the subsurface of Mars?
That's a great question. So one of the key things about so the subsurface brines that are predicted to exist on Mars and the ocean from the Orosai paper, I believe from 2018, 2019, where they believe they detected an ocean beneath the northern pole. A key thing is that it's not just sodium chloride. Like hypersalinity, salinity refers to salt as we know it on Earth. So sodium chloride is the most abundant salt on Earth, whereas on Mars, you've got a lot more magnesium salts and the perchlorates. So there are fascinating hypersaline environments that have the presence of other salts. So in Western Sahara, we see that there are also perchlorates present. They're in the minority, but they're there. And within the variable lakes in northern Canada and Spain, as well as some of the incredibly extreme waters in the Dalao region in Ethiopia, we see that there are these varied chemistries where it's not necessarily sodium chloride dominated, it's dominated by magnesium sulfate, magnesium chloride, and it has a very different selective effect. So what we need to factor in is that even though the water is permitted to exist as a fluid, its salinity exceeds that of all known tolerances of life on Earth. And so we need to study environments that have these varied salt regimes to gain an understanding of what are the impacts evolutionarily, and what are the upper boundaries of life on Earth under those varied chemical regimes.
Okay, thank you. And another one is, okay, how do these microorganisms interact with the factors such as the chemical composition of groundwater and temperature, and how do they survive in this environment?
Cool. So in terms of how do microbes survive within their specific environments, as long as there's sufficiently abundant nutrients to allow either waking up. So there's a key variation here. It's possible to survive, but in a dormant state. So survival and growth are very different factors. It's possible for bacillus spores, again, the amazing example of survival, and all bacterial spores to be present in an environment for a very long time, whether that's a NASA clean room or an incredibly salty brine, but they can't grow. Life isn't actually viable in that environment. It's just not being completely destroyed. If there are sufficient nutrients, and that's one of the things that spores actually wait for, is an availability of compounds to allow them to grow. So sufficient carbon, nitrogen, phosphorus to allow replication. They'll wake up from their dormancy, and that will permit them to survive. In terms of how do they interact, if there are minerals also play a key role in survival in natural environments. So you get some microbes that can directly adhere and enhance the dissolution of minerals, so that will free up greater levels of things like sulfur, iron, like in the pyrite that was mentioned in the previous talk. And in terms of temperature, it's simply a question of if you have the adaptations to survive in those environments. It's like the composition of the membranes and the proteins that will allow you to resist damage. Then you can grow, and if you don't, then they die. So a key piece of work when you isolate a novel microbial strain is you have to show what range of salts, pHs, and temperature it can grow in. And most mesophilic, so non-extremophilic microbes, quite a lot of them can't grow above 40 degrees centigrade. So you have to find that temperature they can't grow at. And yeah, if they can't grow at that temperature, they will simply either remain in a dormant state or die.
Well, connected to this, do you know how let's say a mesophilic microorganism, if you were to put it in an extreme environment for like an experiment, how does it affect its QS system? So the communication between each bacteria in the colony. Do you know anything about that?
Yeah. I mean, quorum sensing is fascinating, and I mean, when microbes are stressed, even mesophiles, there's something they can do. So biofilms are a very common strategy, and when cells are stressed, and stress can mean a whole variety of things, the quorum sensing will, in some cases, if they can survive under that set of conditions, quorum sensing will become incredibly active, and they will trigger biofilm formation. So this is how microbes block drains, because the flow of fluid is stressful. This is how microbes survive antibiotic exposure. If we think about a harsh chemical environment, if you're tuberculosis, that being exposed to an antibiotic is incredibly stressful, because that's an upper chemical gradient. That's a compound that can completely compromise your cell structure. So being able to form biofilms is one QS or quorum sensing dependent survival strategy. Again, it comes down to upper limits of survival. And it's something that we see in the space exposure experiments. So quite often you'll get biofilms formed in response to space exposure. And it's less a case of, this hand is the things being exposed to space. These cells will die, and these cells are shielded. So it's not so much that these cells are adapted to the space conditions, it's that they're not being exposed to them. And we see that in microbes, we see that in eukaryotes, where having this outer layer that dies and is protecting to a limited extent the internal cells is a viable strategy. It's a limited strategy because eventually you'll run out of nutrients. And so, for example, if you were to take a friendly lab strain of E. coli and dip it into a salt solution, again, it would probably die. But if you were to simply put it into its standard medium and put the salt concentration up slightly, you would maybe start to see formation of biofilms or other stress factors.
So connected to this again, because I'm curious. Well, how, in what way would extremophiles create biofilms? In which environment? Is it again when it's beyond their standards, or is there another thing that comes?
If there's flow of liquid, I mean, there's not in Color Peak, but in some of the other springs on Axel Heiberg Island, and also in some of the outflows from hot springs in Iceland, you get what's known as streamers. So it's like, it looks like string, and it's just microbial biofilm. It's having a great time. Like, they don't mind the cold, they don't mind the salt, but by forming these streamers, they can better access the water. And it actually, you can pick these up. They're clumps of microbial cells. So in some ways, it's a valid way of accessing greater nutrients. I was recently on fieldwork in the Nwetwe Pan in Botswana, and again, we found these incredibly thick biofilms. It's just another way for microbes to grow. We don't know why that was specifically triggered in Nwetwe, but what we think is it was potentially we'd come just after there was a lot of water, so the microbes were like, brilliant, let's grow lots, and then it was receding, and then they were like, okay, and formed the biofilm as an attempt to try and persist.
Okay, that's great. And we have a lot of questions very connected to like biofilms. But I have to keep going from the chat. And yes. Are there any areas on Mars where halophiles are particularly detected, or do you think there is a salt lake formation on Mars?
So far, there has never been detection of life on Mars. There was a Martian meteorite that people thought that there was something that resembled a fossilized microbe on. The problem we have as a non-chemist is one of chemistry, which is it's very hard to tell minerals that look exactly like fossilized microbial cells from microbial cells that are fossilized, and it's possible to make synthetic microbial fossils. So that's what that was. So were life ever present on Mars, the last life on Mars would have been halophilic, because as the waters became increasingly desiccated, that didn't happen overnight. That water was dried out over a very extended period of time, relevant for evolutionary selection pressures. There are some studies that suggest that microbes can survive within salt crystals on the scale of millions of years. That's, of course, very complicated to work out, because there would be more modern halophiles living on the outside of the salt crystals. So it's really hard to work out how much we can extrapolate from that. But there are salty bodies of water beneath the surface of Mars. What's actually fascinating, there was just a study that came out on some of the Perseverance samples, and it's effectively the water activity in what way round was it? It was basically the fluids can either be liquid, and the salt concentration is low enough that the water activity is, and water activity high enough that microbes could potentially grow, but the temperature is way too low. Or the temperature is high enough for cells to be able to grow, but the salt concentration means that nothing would be able to grow in those fluids. So it's fascinating. But yeah, they do detect periodic upwellings of fluid on the surface of Mars. And again, this is going to be something like a calcium sulfate enriched fluid upwelling, as opposed to just water. And it cannot be sustained as a consequence of the low atmospheric pressure, as well as all of the other things that make Mars incredibly hostile, the surface of Mars at least.
Okay, thank you. And next question is, what have you learned through your antibiotic resistance researchers on the limits of extremophiles?
Cool. So this is a new strand of research that the Open University is funding me to do. So what we've done is sourcing halophiles from the UK, because of course there's all kinds of ethical issues of trying to study things for potential pharmaceutical production if it would belong to another country. What we found is that halophiles do have genes for biosynthetic production of potential antimicrobial compounds. What we're doing is we're buying in what's known as the escape pathogen. So that's an acronym. Each letter corresponds to one of the 6 most relevant and antimicrobial resistant pathogens in terms of hospital infections. And what we're going to do is we're going to take these halophiles that have these candidate genes, and we're going to screen them in the same way that you take a soil streptomyces and see if there's production of any compound. What's really nice with halophiles and taking this approach to researching production of novel antimicrobials is microbes don't express all of their genes at the same time, and sometimes we have to grow them under specific stress conditions to get them to express those genes. With halophiles, we can do that in a very streamlined manner because we can just grow them on a salt gradient, and then if we go 5 molar salt, 4 molar, 3, all the way down to near freshwater concentrations, we have 5 perfect environments for expressing completely different gene clusters. So yeah, our leading theory is that salt exposure and stress will trigger those different genes to be expressed, and yeah, that's where that project is. So it's very much in its infancy, but we already have a few pet microbes that we're hopeful and we'll be testing in the next couple of months.
I like the saying pet microbes.
Yeah,
That's
well,
great.
I mean, you do come to think of them that way. You spend a very long time looking at them on a plate.
And I have another question. It took a lot of actually, people are really curious. We have a lot of questions. That's great. And this one is, do we have any specific understanding of what types of extremophilic microorganisms can survive under extreme conditions such as extreme cold, radiation, and low pressure on the surface of Mars?
That's a great question. So we have understandings of extreme cold and salinity from environments like Axel Heiberg, also some of the Arctic and Antarctic studies, so things like the McMurdo Valley. In terms of radiation, so there are polyextremophiles, so there are organisms that can survive some of these stresses, but to terrestrial standards, if that makes sense. So nothing approaching what we see on Mars. So the Wadsworth-Kell study I mentioned, where they show the bactericidal effects of the chlorates being hit by UV radiation and the awful chemistry that occurs as a result of that, the fact that just rips spores to shreds, is the same reason that when we consider phototrophy, so cyanobacteria and things like that, we consider that maybe in an early Martian context, but definitely not today, because anything that needed a photosystem would, again, just be ripped apart as a consequence of the radiation and the chemistry. Low pressure is a really interesting one because there are some low pressure environments on Earth. So if you go up the Andes, there's slightly lower pressure there, but nowhere near approaching what is present on the surface of Mars. We don't have like tens of millibars on Earth. So to that extent, we're actually limited in our understanding of adaptations to low pressure, because they don't really exist on Earth. And to investigate that, we need very, very specialized lab equipment. That's not to say that cannot be investigated, but there isn't really an understanding as to how extremophiles would survive under hypobaric conditions in the same way there is for hyperbaric, like we see on the bottom of the ocean floor, because it simply isn't really present on Earth. So again, yeah, there are some extremophiles that can tolerate some of those specific stresses, but nowhere to the extent that they're present on the surface of Mars.
Okay, we don't have the enough how can I say this? Enough environment to replicate Mars, so it's not like we cannot test this almost really exactly.
We can test this in the laboratory, and we can see how cells respond. And there are some studies that have looked into this that have shown cells, some microbial cells are more resistant to hypobarrier than others. I'm stressing the O because it sounds exactly the same as hyperbarrier, which we have a much greater understanding of. And there are some interesting interactions with chemistry. So most likely, if you can survive in low pressure, you're also halophilic, because if you're in a body of fluid, well, fluids evaporate under low pressure. The boiling point is massively reduced, and so the fluid you're in will just become more concentrated by default. There are also, there's potential for maybe evolutionary experiments. So we do know about some microbes from growing like thousands of generations in the lab, how they change. There's a microbe called Vibrio natrogens, which is a halophile. It's a Vibrio, but it's not pathogenic, and its replication time is 10 minutes. So it's the fastest growing organism known to Earth. It's so fast growing that even between different labs that work on it, there are completely different strains. They started with exactly the same one, but after a few just years of working with it, it's replicated enough and undergone genetic drift, random variation in the genome, deletion, because it's grown under different conditions, that they're separate strains. So there are ways to investigate how do cells respond to that stressor. But yeah, in the same way, to get the level of ionizing radiation exposure that's relevant to the surface of Mars or the icy moons, we need labs for that. We can't do that. Thankfully, we can't do that anywhere just on Earth.
Yeah, thankfully. And the next question is from me, actually, because I have been reading a couple of books from the Open University. So I'd like to know what are the advantages and opportunities you have gained through studying and teaching astrobiology in the Open University, and would you recommend students to apply for their studies on astrobiology to there?
That's a great question. So the Open University is an interesting one in the UK because we're a university of the air. We do all remote teaching for undergraduate and master's students. We do have PhD students physically on campus. So I've worked at the Open University in the astrobiology group for 7 years now. And so we're one of the largest astrobiology groups in Europe. We are always looking for further funding and new projects. So if you're interested in astrobiology and are hoping to do a PhD or a postdoc position in the UK at some point, I'd say definitely consider contacting us. There's also the Astrobiology Society of Britain, which has a really helpful map. It's slightly out of date, but it's quite a helpful map, and it shows all of the astrobiology groups in the UK. And that's a very useful resource. So within the Astrobiology OU group, we don't just do microbiology. There's also geology and modeling. So yeah, I'd if you're interested in the UK, I definitely recommend going to the society as a resource for where else there is, because as with funding everywhere, we try to have PhD recruitment every year, but it's at the whims of academia.
Well, I found really helpful books from the Open University, like online books, and they were great to understand.
There's a couple of great texts. Yeah, so Charles Cickel, who runs the UK Centre for Astrobiology in Edinburgh, used to work at the Open University. We also at the Open University, so the director of the group, Professor Karen Olsen-Francis, has done a lot on expanding astrobiology at the Open University. There's also a lot of instrument development, so not from the astrobiology perspective, but from the space explorations perspective. The OU has a very strong track record in instrument development. So it's a great institute.
Yeah, it's great. Well, I'm thinking of it. Thank you for the information. And okay, so another question is, do we know or are there any theories about which extremophiles can survive on which planets?
So there's also a lot of work that's done with regards to potential habitability of the icy moons, and as was described in the previous talk, so there's also the compounding issue of pH. So you would expect that any organisms that did survive there or could survive there would have to be alkalophilic as well as halophilic, as well as potentially having to survive quite extremes of temperature and radiation, depending on where exactly in the ice shell or the ocean that they were. In more of a Martian context, it's more, as one of the previous questions summed up, it's the consequence of pressure, radiation, and salt. Although, interestingly, we have to consider maybe there is at least the possibility that life once existed on Mars, but is now extinct as a consequence of the depletion of the atmosphere. I mean, if the same thing happened on Earth, that would be an extinction level event for Earth, which fortunately, the exact same thing won't happen because we're a different size to Mars and our core isn't going to stop spinning. But yeah, it's also possible that less extremophilic organisms would have been required under an early Martian conditions before it started to lose the water. Maybe mesophilic growth would have been viable, but yeah, not able to persist into what we would consider. It's probably something on the scale of the Great Oxidation Event on Earth. Before there was masses of oxygen in the Earth's atmosphere, most life would have been anaerobic, and it would have been everywhere. And then all of a sudden, there's masses of oxygen. Most life on the surface would have died. The anaerobes persisted, and there would have been some oxygen-tolerant organisms, and there are precursors. But yeah, it's interesting to think about it from the perspective of you don't necessarily need extremophiles for all extraterrestrial environments, just within our solar system.
I have to move on a little quickly. So, as you mentioned, different extremophiles need different extreme conditions. Is there an extremophile that needs conditions like low, high gravity, high carbon dioxide, methane, no oxygen like on Mars, Titan?
That's really cool. So there are experiments that are done under low gravity. So you can simulate that in the lab, and you can also not simulate that. You can also get that through spaceflight missions. So there are some interesting evolutionary experiments, again, looking at how the microbes shift, even from a morphological standpoint, like do the individual cells change shape? And there are some facilities in the Netherlands to simulate hypergravity, so incredibly high levels of gravity. But again, these aren't environments on Earth. I mean, I guess in the atmosphere we have lower levels of gravity, and we do have microbes circling around in the atmosphere. But yeah, the high gravity point is really interesting because, again, it's something that doesn't exist naturally. In terms of the high CO2, we do have environments on Earth where there are high levels of natural outgassing. And so we do have microbes that are naturally adapted to those higher levels of CO2 and are anaerobic. So you get methanogens that are isolated from those types of environment. We also now know about anaerobic methane oxidizing archaea. So this is one of the exciting things about being a microbiologist is we're constantly having our assumptions challenged. So it was known for decades that bacteria can grow methane, they need oxygen because of the enzyme that they use. Cool. There's this entire group called the ANME, so ANME. So I actually can't recall what that is an acronym for, but they're the anaerobic methane oxidizing archaea. It wasn't known that archaea could do that. They do it in the absence of oxygen. None of them have been isolated in pure culture, but we know that they are present in multiple environments. So canals in the Netherlands, there's tons of them, and they're studied extensively. They're very hard to get into pure culture is the reason we don't have them, but they're anaerobic, they grow on methane. They're found in an array of unusual environments. So this is something that we didn't previously know about at all. And then it's just massively expanded our understanding of how those metabolisms could function. So whilst we don't have things that match all of these specific parameters, it's also interesting to consider, could there be an organism, could there be a metabolism that doesn't exist as we know, but could potentially be viable? And it may turn up that something with that specific metabolic pathway is viable. There's only so many electron transfer pairs.
Yeah, well, there's always something you don't know in biology.
Always.
And okay, actually, this question is really interesting because I want to know your idea on this. What do you think about similarity in mineralogical and geological content between the Jezero crater and Lake Salda in Turkey? Could this be a favorable environment for extremophiles to evolve? So what do you think on that? And I could tell you about a little bit about Lake Salda, but what do you know about it?
Yes, if you could tell me a little bit about Lake Salda, that would be great.
Yeah, I thought about that, so I did a little research. So basically, Lake Salda is thought to have eroded from large mounds called microbiolites, and they think hydrothermal magnesite sediments may be similar to carbonate minerals detected at Jezero. And this is from NASA's website, so I'm being really safe. And so basically, it says these structures themselves are good indicators that microbes were once active. So researchers will be looking for signs of these rocks in the Martian crater. So this is what I found, basically. And what do you think about that?
Well, I mean, absolutely is the answer. So even ignoring biology briefly, the mineralogical analysis and the geological context of studying that lake has huge implications for our ability to understand these Martian environments, and that's why it's so important to study them. I think that it can definitely be classed as an analog if incredibly similar mineralogies have been found between the two of them, and working out what are the similarities and also what are the differences and what are potentially driving those. It's definitely worth further study. I mean, we still don't have a completely unambiguous biosignature as well, because what microbes tend to do is they enhance formation of specific minerals. They aren't exclusively responsible for it. So sulfate reducers make pyrite, but if you have a reducing fluid, probably pyrite would form anyway. So from the geological point of view, gaining an understanding of how the fluids evolve and learning as much as we can about something we can go to and dig up samples and look at under a microscope, I think that's hugely important, because I mean, when Mars sample return happens, it's going to be like 0.5 gram to each nation, and we're going to have to try and do as much with that as we can. So we need to make sure that we're asking the right questions, and studying environments like this is a key part of that. Now, in terms of extremophiles evolving, as I mentioned, it's possible that some of these former waters on Mars wouldn't have been extreme. They would have been, much more clement, just like a normal-ish lake on Earth. But developing our ideas of habitability and these different waters and how they've evolved on Earth and could have evolved on Mars, I think there's lots that can be gained from studying these natural environments.
Thank you. And very connected to this, the next question and the last question is, what is the future of analog environment zones research for Mars?
That's a great question. So, I mean, there were always really interesting environments to study. I think taking this in a slightly different direction than possibly, so I'll answer their context and I'll take it in a slightly different direction quickly. I think that there are lots of analog environments that it's worth going back to that have already been characterized, because technology is always moving on, and sequencing platforms I used during my PhD don't exist anymore because they're too old and we have massive advances. So we can always learn more, and also getting a time series as to, how did the springs in Axel Heiberg evolve? How do the salt flats in Western Sahara evolve? And seeing how shifts in chemistry, we can develop a much greater understanding to how the microbial community and the geochemistry of those environments evolve over time. Time points for extreme environments are incredibly rare, and I think it's important that we develop a greater understanding of that, because viability isn't a single snapshot. Viability is persistent. I think something that should happen and really needs to happen with the future of analog environmental research, the UK doesn't have a lot of extreme environments. That means that we go to a lot of other countries and study their extreme environments. There's a concept in science called parachute science, which is effectively you drop into another country, take a bunch of samples and leave, which is a completely unethical approach to research. So what we always do is we will only enter into a country with which we have a working collaborative relationship with a research institute that's already working on those environments. So BUST in Botswana, we didn't just drive around the pan on our own. We specifically organized fieldwork trip with other researchers, with students from BUST, with lecturers from BUST. And I think that from certain nations that have a history of parachute science, it's important that when we're engaging with countries that host these analog environments, these incredibly important environments, that we're engaging with them on an equitable footing, that we're engaging with the scientists in that country, and we're not just doing, like the British Museum did for hundreds of years of just going to countries, taking things and leaving. Because we get papers out of that. We do very well out of that, because you have this really prolific academic career, but then when the scientists within that country want to publish, they'll find it harder. And so I think that astrobiology as a field is starting to move in that direction and needs to keep moving in the direction of working with all of the nations with which we interact as partners, and ensuring that we're not simply profiting from the resources in a way that people within the country maybe don't get a chance to.
Well, we can reasonably say that like analog environments help researchers all around the world reach other parts of the world and ethically and nicely, basically.
Yes.
Well, thank you so much for this session. And we had a lot of questions. It was so nice to see all these questions, and you are very knowledgeable. It was so nice to learn so much from you. Thank you so much for coming. And maybe in the next years we'll see you again, because we do this
thank you very
conference
much.
every year. Also, I'd like to say that I've noticed that you added our logo in your slides.
Yeah.
And I would like to thank you, because that's so nice. That's like such a great thing to do. Thank you.
Of course. Well, thank you very much for the invitation. It's always great to get to take part in these types of events. So thank you.
And thank you so much, of course. Always you're welcome. And have a nice day.
Take care.
Now the English sessions will be moving on in the next hours, but right now we're going to have a question Q&A contest with some also reward as well. So, and that's going to be in Turkish, so you can be in the next session for the rest of the conference. Arkadaşlar, şimdi ödüllü soru cevap yarışmamız olacak. Hepinizi davet ediyorum. Kısa bir aradan sonra sizinle birlikteyiz ödüllü soru cevap yarışmamızla.
Merhabalar arkadaşlar. 4. Uluslararası Agenastro Astrobiyoloji Konferansı'na hoş geldiniz. Umarım şu zamana kadar olan bütün sunumlarımızdan çok büyük keyif almışsınızdır ve sizin için çok eğlenceli geçmiştir. İsterseniz şimdi bugün Astro Kafa'nın sponsoruyla yapmış olduğumuz ödüllü bilgi yarışması etkinliğimiz olacaktır. Sorularımız bildiğiniz üzere böyle çok ciddi sorular olmayacak. Geçen senelerki gibi geçen senelerde biraz daha ciddi sorularımız. Bu sefer birazcık daha eğlenceli sorularımız olacak ve sürpriz dolu sorularımız olacak. İsterseniz sorularımıza geçelim ve aynı zamanda arkadaşımız olan, ekip arkadaşım olan Zeynep Eş'i de davet ediyorum.
Merhaba arkadaşlar, tekrar ben.
Hoş geldin Zeynep.
Hoş bulduk. Bu kısımda Berra yönetiyor olacak. Berra soruları soracak. Ben biraz bekledikten sonra cevapları söyleyeceğim. Berra'nın dediği gibi geçen sene de esprili sorularımız vardı ama ciddi sorularımız daha yoğunluktaydı. Bu sene tamamını esprili yaptık diyebiliriz.
Çok eğlenceli olacak valla. O zaman alıyorum ekrana. Tamam, aldım. Tekrardan ödüllü bilim yarışmamıza hoş geldiniz arkadaşlar. Geçiyorum ilk soruma. Hangi gezegen kırmızı gezegen olarak bilinir? Bu arada sorularımız 30 saniye olacak. Şu an 20 saniye olacak. 20 saniye açtım. Evet, şu an cevaplamak için
Cevaplarınızı bekliyoruz arkadaşlar.
bayağı
Oldu mu?
cevap
20 saniye.
veriyor şu anda. Sanırım oldu. Ben açmayı unutmuşum bu arada. Tamam.
Tamam,
Oldu.
cevaba geçelim. Kırmızı gezegenimiz Mars arkadaşlar.
Mars çünkü bazen ben okuyamıyorum bu arada. Altı sen okuyabiliyor musun Zeynep?
Evet. Bazen kızgın olabilir ama asla utanmaz.
Bir sonraki sorumuza geçelim. Bir gezegenin yüzeyinde yaşam ararken hangi temel bileşenin varlığını ararız? A. Kahve, B. Su, C. Çikolata, D. Soda. Bir gezegenin yüzeyinde yaşam ararken hangi temel bileşenin varlığını ararız? Kahve ararız arkadaşlar. Kahveye hepimizin ihtiyacı var.
Kahvesiz yaşam yok herhalde.
Bence kesinlikle kahve olmalı. Ben şu an ayakta zor duruyorum.
Aramayız. Çikolatayı arıyorsun yani Dilara. Bence yeterli. Geçelim cevaba. Arkadaşlar tabii ki belli olduğu üzere su. Şöyle ilerleyelim. Su çünkü bu bizim can suyumuz. Berra sanırım dondu. Berra orada mısın? Berra. Berra'nın internete gitmiş. Hemen geliyor. O sırada isterseniz daha cevap vermenize gerek yok arkadaşlar. Cevabı gösterdim. Ben cevabı gösterdikten sonra verdiğiniz cevaplar geçerli olmayacak. Şimdi tekrar açmam gerekecek. Berra gittiği için. Diğer soruya geçelim. Güneş sisteminde hangi gezegenin uydusu olan Europa'da su altında okyanuslar olabileceğine inanılıyor? A. Mars'ın, B. Jüpiter'in, C. Satürn'ün, D. Uranüs'ün. Burada asıl sorduğumuz Europa kimin uydusu? Evet arkadaşlar, cevap Jüpiter. Çünkü bir yerlerde denizaltı balinası dolaşıyor olabilir diye bir espri yapmıştık. Evet, Berra da geldi.
Tek kahveye ihtiyacımız yok. İnternete de ihtiyacımız var. Özellikle benim.
Berra, ben bir sonraki soruyu sordum. İstiyorsan bundan sonrasında sen devam et. Bir sonraki soruya geç. Arkadaşlar, ben cevap gösterdikten sonra cevap vermeyin. İlk başta verilen cevaplar önemli. İlk 3 kişi sadece ödül alacak. Bilmiyoruz, sürpriz. Ödül ne diye sormuşlar.
Astrobiyologlar hangi tür gök cisimlerini yaşamın potansiyel izlerini aramak için incelerler? A. Asteroidler, B. Yıldızlar, C. Galaksiler, D. Uydular. 20 saniyemiz başladı arkadaşlar. Son 6 saniye. Çok kıyasıya ilerliyoruz arkadaşlar.
Yani genelde de hep doğru cevap veriliyor.
Evet. Evet, cevabını veriyorum arkadaşlar.
Çok hızlı geçtin. Cevap asteroidler.
Asteroidler, çünkü ben okuyamıyorum bu arada ya. Çünkü
Niye?
Burada yazı gözüküyor da okuyamıyorum. Sen okuyabilir misin? Alt tarafı okuyamıyorum.
Çünkü onlar da bir tür uzay taşları. Ekranda tutarsan mouse'unu göremezsin. Mouse'unu farklı bir yere çekersen görebiliyorsun.
Tamam.
Gördün mü?
Bir sonraki sorumuza geçelim isterseniz. Arkadaşlar niye kahvaltı yapmadık? Kahvaltı çünkü çok fazla kişiyi alamıyoruz. Halka açık olduğu için onun belli bir sınırı var. Ondan dolayı olmuyor arkadaşlar.
Evet, bir de Kahoot'ta sürekli cevap veriyorsunuz. Cevapları görüp hileler oluyor.
Ondan dolayı maalesef onu yapamadık. Bir sonraki sorumuza geçelim hemen. Hangi gezegenin uydusu Titan sıvı hidrokarbon denizlerine sahiptir? A. Mars'ın, B. Jüpiter'in, C. Venüs'ün, D. Satürn'ün. 20 saniyemiz başladı arkadaşlar.
Burada da Titan'ın kimin uydusu olduğunu soruyoruz. Bu arada biri demişti ki nasıl puanlayacağız? Evet, şu an okudum. Şöyle olacak. YouTube chat'te sizin cevaplarınıza bakacağız. İlk cevap verenler ve en çok doğru bilenler, en çok doğru bilip en hızlı cevap verenler arasından ilk 3 kişiyi seçeceğiz. Sonrasında Instagram hesabımızda duyuracağız.
Evet. Bu arada süremiz bitti arkadaşlar. Cevaba geçelim o zaman.
Cevabımız Satürn arkadaşlar.
Cevabımız
Gerçekten biraz yağlı görünüyor. Şişmanlığına burada bir atıf var.
Sapsarı aslında ama. Bence çok güzel bir gezegen ve mesela şu an burada yazmıyor ama benim ikinci ismim Zühal. O da Satürn demekmiş.
Evet.
Çok güzel gerçekten. Benim en sevdiğim gezegen.
Orada gerçekten Satürn'ün rengi de çok hoş.
Evet, kesinlikle. Dünya dışı yaşam ararken hangi tür gezegenler daha fazla ilgi çeker? A. Küçük ve kuru gezegenler. B. Büyük ve gazlı gezegenler. C. Hızlı dönen gezegenler. D. Çok renkli gezegenler.
Ya bence bu biraz böyle hani çelişkili bir soru bence. Yani ben başkalarına sorduğumda bilemediler. Öyle diyeyim. Evet, farklı cevaplar gelmeye başladı bak.
Evet, burada kırılıyorsunuz arkadaşlar.
İyi ki böyle bir soru eklemişiz. Yoksa kimseyi seçemeyecektik.
Evet, birazdan tamam arkadaşlar. Süremiz
Bence
doldu.
yeterli. Küçük ve kuru gezegenler arkadaşlar.
Küçük ve kuru gezegenler, çünkü kimse okyanusta yüzmeyi beklemiyor.
Hızlı dönen gezegenlerde yaşam aramamız çok mümkün değil. Çok fazla katılaşmış olur. Büyük ve gazlı gezegenler hem büyüklüğünden dolayı hem de gaz yoğunluğundan dolayı çok mümkün olmaz. Çok renkli gezegenler dediği de yani büyük ihtimalle ilk evreler veya son evreler oluyor. Onlar da olmaz. Küçük ve kuru gezegenler aslında bizim en çok dünya dışı yaşamı aradığımız gezegenler.
Bir sonraki soruma geçiyorum. Hangi gezegen gezegenlerin kralı olarak bilinir? A. Jüpiter, B. Mars, C. Venüs, D. Neptün. Burada şöyle bir info vereyim size. Asteroid kuşağının hangi gezegeninin oradan geçtiğini biliyorsanız eğer bu sorunun cevabını da bilirsiniz arkadaşlar.
O daha zor oldu. Bence oradan gitme. Büyüklükten gidelim.
Böyle büyük, böyle bütün yer çekimi, bütün alanı böyle kendi etrafında döndüren bir gezegen olduğunu düşün.
Güneş sisteminin en büyüğü.
Öyle bir kral.
Şeyden de düşünebilirsiniz. Yunan tanrısından. O da kral. Tanrıların tanrısı diye biliniyor hatta.
Benim kopyalarımla.
Evet. Bu akşam Haşim, bu arada Astro Kafa'nın kendisi kurucusu, bu akşam gökyüzünde de görülecek bir gezegen dedi. O zaman cevabımıza geçiyorum artık arkadaşlar.
Jüpiter arkadaşlar.
Jüpiter çünkü diğer gezegenlere göre biraz daha kilolu. Evet, bir sonraki sorumuza geçiyorum arkadaşlar. Dünya dışı yaşam arayışında hangi faktör en önemlidir? A. Uzay gemilerinin hızı. B. Yer çekimi. C. Sıcaklık. D. Kedi fotoğrafları. Bence kedi fotoğrafları arkadaşlar.
Ulan dur geleyim ben de trolleyeyim azıcık.
Evet, cevaplar geliyor. D diyenler olmuş. Gerçekten. Evet, herkes kedici arkadaşlar. Hepimiz kediciyiz. Evet, süremiz artık...
Çok doğru bilmiş zaten bence. Bak, fazlasıyla C var çünkü.
Evet,
Sıcaklık
C,
arkadaşlar.
Sıcaklık çünkü
Soğukluğa dayanamayan uzaylılar varsa
onları battaniyelerle karşılamalıyız. Öyle yazılıyor sanırım. Tamam, evet. Bir sonraki sorumuza geçiyorum arkadaşlar. Hangi gezegenin uydusu olan Evet, bu nasıl okunuyor ben de bilmiyorum. Aslında hocamızın da var.
Enceladus.
Su püsküren jeotermal aktivitelerle ünlüdür. A. Mars'ın, B. Jüpiter'in, C. Uranüs'ün, D. Satürn'ün. Arkadaşlar ilk sunumumuzu izlediyseniz bunu bilmeniz gerekiyor. Evet, ilk sunumu izleyenler bu soruyu bilecek.
Çok fazla yanlış cevap var. İlginç. Bence cevaba geçelim.
Sunumumuzu izlememişsiniz. Yakaladık
Cevaba
sizi.
geçelim bence Berna. Arkadaşlar tamam. Cevap D. Satürn'ün.
Satürn'ün çünkü
içini dökme isteği var. Bunu da yine sunumdan anlayabilirsiniz. Hidrotermal ventlerle su püskürtüyordu.
Arkadaşlar nasıl bilemediniz bu soruyu? Neyse bir sonraki soruya geçiyorum arkadaşlar. Göktaşı düşerken hangi sesi çıkarır? A. Bir şey düşüyor. B. Süpernova stilimle iniyorum. C. Çok yaklaşan bir uzaylı saldırısı. D. Ah anneciğim bana sıcak çikolata getir. Evet, hangi sesi çıkarır arkadaşlar? Bir arkadaşımız viu dedi. Bence kesinlikle öyle. Evet. Evet, herkes farklı cevaplar veriyor.
Ama daha çok A ve B'ye yoğunlaşmış herkes. Bu çok güzel.
Evet, komutan Logar. Kendisi benim sınıf arkadaşım olur bu arada.
Yasin'inki de çok güzel. Ben göktaşı düşüyorum.
Ben göktaşı düşüyorum.
Arkadaşlar cevabımız süpernova stilimle iniyorum. Ne de olsa bir gösteri yapması lazım inerken.
Evet arkadaşlar, bir sonraki sorumuza geçiyoruz o zaman. Dünya dışı bir gezegendeki yaşam formu insanlarla iletişim kurmak için hangi dili kullanmayı seçer? A. Esperanto, B. Uzayca, C. Emoji ikonları, D. Klingonca. Klingonca ne ya?
Klingonca. Aa. Berna üzdün beni.
Klingonca ne ya?
Hemen sana film etmemiz lazım.
Bilmiyorum. Emoji ikonları. Ben de bilmiyorum arkadaşlar.
Bu arada bu esprili cevaplar dışında geçen seneki konferansa bir tane hocamız davet edilmişti. Onda uzaylılarla iletişim için hangi dili kullandığımızı göstermişti.
Bundan bahsediyorsun. Evet.
Aynen. Süpermen. Süpermen'in dili.
Bilmiyorum. Üzgünüm. Bilemedim. Evet. O zaman cevabımıza geçelim isterseniz arkadaşlar.
Cevap Klingonca arkadaşlar. Tabii ki de yani.
Çünkü birazcık klasikleşmiş bir seçenek. Hiçbir bilgim yoktu arkadaşlar. Beni buradan atın. Vallahi bilmiyordum. Evet. Bir sonraki sorumuza geçelim o zaman. Uzaylılar dünyayı ilk ziyaretlerinde ne tür bir hediye getirirler? A. Uzay çiçekleri. B. Yenilebilir enerji teknolojisi. C. UFO belgeseli DVD'leri. D. Evrenin en sevilen çikolatası. Bence çikolata. Kedi maması. Evet, kedi mamasını niye yazmadık arkadaşlar ya? Kedi maması.
Şeyi zaten şıklardan birini yanlış yazmışız. Yenilebilir değil, yenilenebilir.
Ben de onu diyorum. Nasıl
Enerjiyi niye yiyoruz biz?
enerji yenilir arkadaşlar?
O da uzaylı teknolojisi demek ki.
Yeniliyorsa okey. İzmit
B
pişmaniye.
ama C şıkkını çok sevdim diyen var. Yenilebilir enerji varsa o da kedi maması sayılır
Bence
demiş.
kedi direkt. Kedinin kendisi
Ben
olsa.
yesem yerim enerji demiş. Peki.
Evet, cevaba
Arkadaşlar
geçelim.
cevabımız çikolata tabii ki. Klasik bir hediye değil mi? Bu da her zaman hoş bir jest. Yani Almanya'dan bile hep çikolata getiriyoruz.
O zaman bir sonraki sorumuza geçelim arkadaşlar. Gezegenler birlikte bir parti düzenlediğinde hangi oyunu oynamayı tercih ederler? A. Uzay Monopoly. Bence Monopoly. B. Plüton'un unutulmuşluğunu hatırlamak. C. Yıldızlar arası karaoke. D. Satürn'ün halkaları hakkında gizli sırlar oyunu.
Arkadaşlar Berra o cevabı verdi diye cevapları biliyormuş gibi aynı şey söylemeyin. Az önce çikolatayı bildi ama ondan öncekini bilemedi biliyorsunuz değil mi?
Ben bu arada
Filmden-
cevapları hiçten bilmiyorum. Hazırladım ama hani o kadar hızlı baktım ki hiçbir fikrim yok yani. Ben kendi cevabımı söylüyorum. Bence Monopoly'dir.
Güzel
Ama
oyun
B
olur
de
aslında.
B de güzel. Bence B bu arada. Plüton'un unutulmuşluğunu hatırlamak. B.
Bence cevaba geçelim. Herkes fazlasıyla verdi
Evet.
çünkü. Arkadaşlar cevap Yıldızlar Arası Karaoke.
Çünkü galaksi
Bence-
genelinde hepimizin sevdiği bir aktivite.
Bence öyle. Ben Kore draması seven bir insanım. Karaoke de severim. Soruları ben hazırladım. Bu çok güzel bir cevap evet.
Bir sonraki sorumuza geçelim arkadaşlar. Bir uzaylı kıyafeti satın alırken hangi kriterler en önemlidir? A. Rengi. Çünkü uzaylı da modaya uymak önemlidir. Kesinlikle her yerde modaya uymak önemlidir. B. Yıldız sayısı. C. Sınırsız cep saklama alanı. D. Yer çekimi karşıtı güzellik kremi içeren. Nasıl bir cevap ya? Güzellik kremi.
Krem akıyor demek ki ya çekimi karşıtı ya. Akmayacak.
Anlamadım vallahi. Ben bile anlamadım. Cep sayısı C.
Bence burada herkes hemfikir diyeceğim ama erkekler hemfikir. Erkekler sınırsız cep istemiş. Kızlar ya modanın rengine gitmiş ya yıldız sayısına gitmiş.
Bence de. Yer çekimi karşıtı kremim olmazsa olmaz. Alaka.
Bir önceki sorunun cevabı galiba.
Evet o bence de. Geç geliyorsun Umut.
Geliyor.
Umut biraz hızlı gel. O zaman isterseniz cevabımıza geçelim arkadaşlar.
Sınırsız cep saklama alanı arkadaşlar.
Çünkü uzaylı
Yani ben-
cep telefonlarına ihtiyaç duyulabilir.
Sadece
Ama
o
çok-
değil. Cep her şeye yarar yani. Uzayda en gerekli şey bence kıyafetler. Bilim kurgu izleyenler bilir.
Evet geçiyoruz. Dünya dışı bir restorana sipariş verirken hangi yemeği istemek en güvenli seçenektir? A. Mars'ta yetiştirilmiş muzlar. B. Neptün'ün buz salatası. C. Venüs'ten gelen sıcak lav çorbası. D. Uranüs'ün atmosferik tatlıları.
Tolga atmosferik tatlıları tercih etti. Biri muzu seçti. Tatlılara rağbet var. Lav çorbası da rağbet görmeye başladı.
Bence muz ya. Buzlu salatası. Bence muz arkadaşlar. Yapıyoruz çünkü. Domates falan yetiştirdik ay toprağıyla. Öyle bir şey
Tolga
hatırlıyorum.
tatlı çekiyor canım şu an çünkü demiş. Sanırım o yüzden cevabını D verdi. Atmosferik tatlı yemeyen de ne bileyim. Patates varsa muz neden olmasın?
Bence de ben de öyle
Yok
düşünüyorum.
arkadaşlar.
Ben
Bu
de
arada.
şara gibi düşünüyorum.
Muzlar.
İsmi çok güzelmiş bu arada arkadaşımızın. Evet isterseniz
Cevap
cevabımıza geçelim.
arkadaşlar Mars'ta yetiştirilmiş muzlar. Muzları
Yani.
kim sevmez ki? Herkes muz sever.
Yani, bence de. Evet bir sonraki sorumuza geçelim.
Bitti.
Ah,
Bu son
olmamış,
sorumuzdu.
bitmiş.
Ama Neptün'ün buzlu salatasını denememiş olan var mı, şaka mı diye de son bir yorum okuyalım.
Aerobik yıkanmış kahve. Kim o?
Hilal.
Katılımınız için çok teşekkür ederiz arkadaşlar. Çok eğlenceli bir sunum olduğunu düşünüyorum. İnşallah eğlenmişsinizdir. Kazananları biz daha sonrasında değerlendirme yapıp sizlere MBG Türkiye Instagram hesabımızdan yayınlayacağız arkadaşlar. Ama umarım kayıt olmuşsunuzdur herkes. Çünkü size ulaşmamız için kayıt olmanız gerekiyor. Lavlı çorba içimizi ısıtır. Uzay soğuktur
Uzay
şimdi.
soğuktur
Doğru
şimdi.
diyorsunuz. İçimiz soğudu. Espri. Zeynep, istersen sen de son cümlelerini söyle arkadaşlarımıza.
Teşekkür ederiz arkadaşlar katılımınız için. Bir sonraki session'ımız, Türkçesini unuttum bir an. Bir sonraki sunumumuzda Bruno Hoca bizlerle olacak.
Saat 5.20'de başlayacağız arkadaşlar. 20
Evet.
dakika aramız olacak.
Hı hı.
Bu arada şunu da eklemeyeyim. Farklı bir linkten devam edeceğiz arkadaşlar. Bu linkimiz
Evet.
sonlanmış olmakta.
Hı hı.
Bruno hocamız da saat 5.20'de tekrardan konferansımıza devam ediyor olacağız. Çok teşekkür ederiz arkadaşlar her şey için.
Görüşmek üzere arkadaşlar.
Görüşmek üzere arkadaşlar.